Hello, friends. Welcome. I'm really glad you're here. To begin, I'd love to hear where you're watching from and what the clock says for you right now. It's amazing to see people gathering here from so many places at all sorts of hours. Thank you also to those who've subscribed and supported the channel by liking the videos. It means more than you know, and it helps others find their way here too. If you have suggestions for future topics, please share them in the comments. Now, let's begin. Chapter 1. Understanding Bacteria. Basics of Microbial Life. Bacteria, those microscopic entities that exist in every environment, are truly remarkable in their diversity and adaptability. They are single-celled organisms, so small that millions could fit on the head of a pin. Despite their size, bacteria play an essential role in the world around us. Imagine the vastness of the oceans, the richness of soil, and the air we breathe. In each of these spaces, bacteria flourish, forming the very foundation of life. These microorganisms are characterized by their simple structure, typically lacking a nucleus and other membrane-bound organelles. Instead, their genetic material resides in a single circular strand of DNA. This simplicity belies their complexity, as bacteria possess a remarkable ability to thrive in various environments, from the depths of the ocean to the arid deserts. They can be found in the most inhospitable places, such as hot springs, where temperatures soar, or in the frozen tundras, where life seems almost impossible. Bacteria reproduce through a process known as binary fission, a method that allows them to replicate quickly and efficiently. Imagine one bacterium dividing into two, then those two dividing into four, and so on. This exponential growth enables bacterial populations to swell in a short period. Conditions such as nutrient availability and temperature can influence this rate of reproduction. Under ideal circumstances, some bacteria can double in number within just a few hours, creating thriving colonies that are visible to the naked eye. This rapid growth is not without its challenges. As populations increase, resources may become scarce, leading to competition among bacteria. They have evolved various strategies to survive in such competitive environments. Some produce substances that inhibit the growth of neighboring bacteria, while others form biofilms, complex communities that provide protection and enhance survival. Biofilms can be found on surfaces ranging from natural environments like riverbeds to man-made structures such as pipes and dental equipment. Bacteria are not just simple organisms, they are vital players in the ecosystems they inhabit. Their roles are multifaceted, contributing to nutrient cycling, decomposition, and even the formation of soils. As decomposers, they break down organic matter, recycling nutrients back into the ecosystem. This process is crucial for the health of our planet, as it ensures that life can continue to flourish. Imagine a fallen tree in a forest. Over time, it decomposes, and bacteria work tirelessly to break it down, allowing nutrients to be released back into the soil, nurturing new plant life. In addition to their ecological significance, bacteria are also integral to human health. The human body is home to trillions of bacteria, collectively known as the microbiome. This complex community plays a vital role in digestion, metabolism, and even immune function. Some bacteria help us break down food, allowing for the absorption of essential nutrients. Others compete with harmful pathogens, preventing them from taking hold and causing illness. The balance of this microbiome is delicate. Disruptions can lead to health issues, ranging from gastrointestinal disorders to autoimmune diseases. Fascinatingly, some bacteria are used beneficially in medicine and biotechnology. For instance, the discovery of antibiotics, a breakthrough attributed to Alexander Fleming, who noted the antibacterial properties of penicillin, has transformed the treatment of infections. This remarkable finding has saved countless lives. Similarly, probiotics, which are live bacteria that confer health benefits, have gained popularity for their ability to aid digestion and bolster the immune system. As we explore the world of bacteria, it is essential to recognize that not all bacteria are harmful. While some can cause diseases, Many are beneficial and play crucial roles in maintaining health and ecological balance. This dual nature invites us to ask, how can we harness the positive aspects of bacteria to improve our lives? Through research and innovation, scientists continue to uncover the potential of these microorganisms, from developing new antibiotics to exploring their use in sustainable agriculture. The relationship humans share with bacteria is complex and multifaceted. We depend on them for health, yet we also must be cautious of the pathogenic strains that can cause illness. This awareness has led to increased interest in understanding how we can promote healthy bacterial populations within our bodies and environments. As we ponder the intricate workings of these microorganisms, we may find ourselves reflecting on the interconnectedness of life. Bacteria, despite their simplicity, are at the heart of many processes that sustain life on Earth. They remind us of the beauty found in the smallest of creatures. Each bacterium plays a role in a larger narrative, one that influences not only ecosystems, but also the human experience. In every drop of soil, in the depths of our intestines, and in the air surrounding us, bacteria thrive, shaping the world in ways we are just beginning to understand. Their stories are woven into the fabric of life itself, and as we learn more about them, we uncover not only the secrets of these tiny organisms, but also the secrets of life on our planet. Bacterial reproduction is a marvel of efficiency and adaptability. In ideal conditions, the rapid division through binary fission can lead to astonishing population growth, allowing them to colonize new environments swiftly. However, this growth often leads to a phenomenon known as density-dependent regulation, where the increasing concentration of bacteria triggers mechanisms that slow their reproduction. Imagine a bustling city where resources begin to dwindle. The same principle applies to bacteria as they compete for nutrients and space. During this competition, some bacteria develop unique strategies to secure their survival. One such strategy is the production of secondary metabolites, which can be antibiotics or toxins. These compounds serve as a defense mechanism, preventing other bacteria from encroaching upon their territory. The discovery of streptomycin, an antibiotic produced by the bacterium Streptomyces griseus, is a testament to the ingenuity of bacterial survival tactics. Nature is a master craftsman, observed the renowned biologist Paul Ehrlich, highlighting how organisms evolve sophisticated methods to thrive. As we reflect on these interactions, it is essential to consider the ecological roles that bacteria play beyond mere survival. They are key participants in biogeochemical cycles, particularly the nitrogen cycle. Through a process known as nitrogen fixation, certain bacteria convert atmospheric nitrogen into forms usable by plants. This transformation is essential for maintaining soil fertility and supporting plant growth. Imagine a vibrant field of crops, nourished not only by sunlight and water, but also by the invisible work of bacteria transforming nitrogen. In aquatic ecosystems, bacteria also play a crucial role in nutrient cycling. They decompose organic matter, breaking it down into simpler compounds that can be used by other organisms. This decomposition process is not merely a breakdown, it is a vital regeneration of resources, creating a continuous loop of life. Without these microscopic decomposers, ecosystems would be overwhelmed with waste, and the cycle of life would falter. Bacteria also contribute to the formation of symbiotic relationships with other organisms, enhancing their ecosystems even further. In the roots of leguminous plants, for example, nitrogen-fixing bacteria form nodules, creating a mutually beneficial partnership. The plant provides carbohydrates to the bacteria, while the bacteria supply the plant with essential nitrogen. This relationship exemplifies the interconnectedness of life, where one organism's survival is intricately linked to another's. Delving deeper into the human aspect of bacteria, we find that our relationship with these microorganisms is both intimate and complex. The human microbiome, teeming with diverse bacterial species, is a dynamic ecosystem that influences our health in profound ways. Research has shown that the composition of our microbiome can affect not just digestion, but also mood, immune responses, and even susceptibility to diseases. The notion that we are more bacteria than human resonates deeply as our microbial companions outnumber our own cells. Consider the impact of diet on the microbiome. A diet rich in fiber, for example, fosters the growth of beneficial bacteria that produce short-chain fatty acids, crucial for gut health. Conversely, a diet high in sugar and processed foods may lead to an imbalance, favoring harmful bacteria. This delicate balance prompts the question, how can we cultivate a thriving community of beneficial bacteria? Engaging in practices that promote gut health may hold the key, from incorporating probiotics to embracing a diverse diet rich in whole foods. In the realm of medicine, the potential uses of bacteria extend beyond antibiotics. Scientists are exploring the concept of bacteriotherapy, where specific strains of bacteria are introduced to combat infections or diseases. The use of phage therapy, which utilizes bacteriophages to target and eliminate pathogenic bacteria, is gaining traction as an alternative to antibiotics. This innovative approach holds promise, particularly in an era where antibiotic resistance poses a growing threat. The exploration of bacteria does not stop at human health. In agriculture, researchers are harnessing the power of bacteria to promote sustainable practices. The use of biofertilizers, which contain beneficial bacteria, can enhance soil health and reduce the need for chemical fertilizers. Imagine fields flourishing with crops nourished by natural processes, fostering both ecological balance and agricultural productivity. As we ponder the significance of bacteria in our lives, we are reminded of the delicate balance of nature. These microorganisms, often overlooked, are integral to the functioning of ecosystems and the health of organisms, including humans. They challenge our perceptions of life's hierarchy, illustrating that even the smallest entities can wield profound influence. In every breath we take and every bite we consume, bacteria are present, performing their roles with quiet efficacy. They are the unseen architects of our world, reminding us of the intricate connections that bind all living things. As we continue to unveil the mysteries of these remarkable organisms, we embark on a journey of discovery that promises to deepen our understanding of life itself. Bacteria's influence extends beyond just their immediate environment. They shape and maintain the intricate web of life that surrounds us. Their presence is a constant reminder of nature's resilience and adaptability. They can survive in extreme conditions, from the salty depths of the ocean to the acidic pools of volcanic springs, showcasing an incredible range of tolerance. This adaptability raises a thought-provoking question. How do these tiny organisms endure conditions that would be inhospitable to most other forms of life? One fascinating adaptation is the formation of spores. Certain bacteria, like Bacillus and Clostridium species, can enter a dormant state, encasing themselves in a protective shell. This spore formation allows them to survive extreme heat, drought, and even radiation. Imagine a tiny fortress, impervious to the ravages of time and environment, waiting for favorable conditions to awaken and thrive once more. When conditions improve, these spores can reactivate, returning to their active forms and continuing their life cycle. The significance of bacteria in ecosystems cannot be overstated. They are pivotal players in the carbon cycle, contributing to the breakdown of organic materials and the release of carbon dioxide back into the atmosphere. This process is essential for maintaining the balance of gases that sustain life on our planet. Through their decomposition activities, bacteria ensure that carbon is recycled, supporting plant growth and, ultimately, the entire food web. Consider the role of cyanobacteria, which are often referred to as blue-green algae. These bacteria are not only photosynthetic, but also crucial for oxygen production. They were instrumental in transforming Earth's atmosphere billions of years ago, contributing to the oxygenation of the planet, which made it hospitable for more complex life forms. Just think about it. The very air we breathe owes much to these microscopic pioneers, who played a foundational role in shaping our environment. In agricultural settings, the relationship between bacteria and plants continues to flourish. Rhizobacteria, which inhabit plant roots, can enhance nutrient uptake and promote plant growth. They help in solubilizing phosphate, a vital nutrient that plants struggle to absorb. This symbiotic relationship exemplifies how bacteria serve as silent allies in food production, working diligently beneath the surface to support the growth of crops that sustain human populations. In terms of human health, the exploration of bacteria therapy reveals yet another dimension of our complex relationship with these microorganisms. Consider the potential of using engineered bacteria to target specific diseases. Scientists are experimenting with genetically modified strains designed to deliver therapeutic molecules directly to diseased tissues. This approach could revolutionize treatment options, offering targeted therapies without the side effects associated with traditional medications. The presence of bacteria in our bodies is a constant reminder of the delicate balance that sustains our health. The microbiome's diversity, composed of hundreds of different bacterial species, is crucial for our well-being. It not only aids in digestion, but also plays a role in regulating our immune system. The idea that our health is intertwined with the health of our microbiome invites us to reflect on how our lifestyle choices impact this invisible ecosystem within us. As we contemplate the significance of bacteria in our lives, it is essential to acknowledge the ongoing research that continues to reveal the many layers of their influence. The study of these microorganisms is not merely an academic pursuit. It is a journey into the heart of life itself. With each discovery, we uncover new ways to harness bacterial potential, whether through innovative medical treatments, sustainable agricultural practices, or ecological restoration efforts. Bacteria remind us of the interconnectedness of all life. They are the unseen architects of ecosystems, the guardians of nutrient cycles, and the partners in our health. As we continue to explore their world, we find ourselves on a path of discovery that deepens our appreciation for the complexity of life and the myriad ways in which even the smallest organisms can shape our existence. In this intricate tapestry of life, the stories of bacteria are woven with those of plants, animals, and humans, creating a narrative rich with interdependence and resilience. As we look to the future, we must consider how we can coexist with these microorganisms, fostering a relationship that respects their role in the grand scheme of life. What more can we learn from these tiny beings, and how might they inspire solutions to the challenges we face? The answers lie just beneath the surface, waiting to be uncovered. Chapter 2. Antibiotics, the discovery and early use. The discovery of antibiotics marks a pivotal moment in the history of medicine, one that emerged from a confluence of curiosity, serendipity, and the urgent need to combat bacterial infections. Picture the early 20th century, a time when bacterial infections claimed countless lives, and the medical community was grappling with the limitations of existing treatments. The quest for a solution led to groundbreaking insights that would change the course of medicine forever. In those days, scientists were honing their understanding of bacteria, the very agents that wreaked havoc on human health. As we learned in our previous chapter, bacteria are ubiquitous, residing in every conceivable environment. However, it was their role as pathogens that sparked a flurry of research aimed at finding ways to inhibit their growth and eliminate their threats. The stage was set for discovery, and the world was about to witness a revolution. The accidental discovery of penicillin by Alexander Fleming in 1928 illustrates the fortuitous nature of scientific breakthroughs. Fleming observed that a mold called Penicillium notatum inhibited the growth of Staphylococcus bacteria in a petri dish. This simple observation, made while he was investigating bacterial cultures in his lab, would soon lead to the development of the first widely used antibiotic. Fleming's words resonate with the spirit of discovery. One sometimes finds what one is not looking for. His insight illuminated a path forward. Penicillin became a beacon of hope in treating infections that once seemed insurmountable. As penicillin became more refined and its applications explored, scientists began to understand its mechanisms of action. Penicillin works by targeting the bacterial cell wall, a critical structure that maintains the integrity and shape of the cell. Imagine a balloon filled with water. The rubber material keeps the water contained. For bacteria, their cell wall is like that rubber, providing essential support. Penicillin disrupts the synthesis of this wall, causing the bacteria to burst under the pressure of internal fluids. This targeted action exemplifies how antibiotics can work selectively, affecting bacteria while sparing human cells. Following the discovery of penicillin, a flurry of research was dedicated to identifying additional antibiotics. Scientists sought to isolate other naturally occurring substances with antibacterial properties. Streptomycin, discovered by Selman Waxman in 1943, emerged as another groundbreaking antibiotic. Waxman's work on soil bacteria demonstrated that certain microorganisms could produce compounds capable of killing or inhibiting the growth of other bacteria. He famously stated, "The soil is a treasure house of antibiotics." This treasure trove of microbial life provided a rich source for new medications, paving the way for a multitude of antibiotics that would soon flood the market. The mechanisms of action for these early antibiotics became clearer as researchers delved deeper into the world of microbiology. Tetracycline, for instance, operates by inhibiting protein synthesis within bacteria. Imagine a factory assembling products. Without the machines that create these goods, production halts. Tetracycline interferes with the bacterial ribosomes, the very machinery that constructs proteins essential for bacterial survival. By disrupting this process, the antibiotic effectively stifles the growth of bacteria, allowing the immune system to catch up and eliminate the infection. The impact of antibiotics on public health cannot be overstated. With the advent of penicillin and its successors, a new era dawned. Bacterial infections that once claimed lives now became manageable, and the mortality rate from infectious diseases dropped dramatically. The introduction of antibiotics transformed surgical procedures, making previously high-risk operations safer. Patients who would have succumbed to infections like pneumonia or sepsis found new hope for recovery. The triumphs of antibiotics also brought forth profound societal changes. As diseases were brought under control, life expectancy began to rise, and public health initiatives flourished. Consider the challenge of tuberculosis, a disease that once ravaged populations. With the introduction of streptomycin, the tide began to turn. The World Health Organization reported that incidence rates fell and lives were saved. The ability to treat and cure infections allowed for a shift in focus toward chronic diseases and preventive care. As we reflect on the remarkable journey of antibiotics from discovery to early use, it is essential to acknowledge the challenges that accompanied these advancements. The rapid proliferation of antibiotics led to the emergence of resistant bacteria, raising concerns that echo in modern medicine today. The very organisms that were once vanquished began to adapt and develop resistance, creating a new set of obstacles in the fight against infections. The story of antibiotics is one of hope interwoven with caution. The early successes ignited a wave of optimism, but also serve as a reminder of the need for responsible use. As the world embraced these powerful tools, it became crucial to understand the delicate balance between their effectiveness and the potential for resistance. In this context, we find ourselves at a crossroads. How do we maximize the benefits of antibiotics while safeguarding their efficacy for future generations? The answer lies in a combination of education, stewardship, and ongoing research. Just as the pioneers of antibiotic discovery forged a path through uncharted territory, we too must navigate the complexities of antibiotic use in a world that constantly evolves. The early use of antibiotics reminds us of the brilliance of scientific discovery and the profound impact it can have on humanity. Each antibiotic tells a story of exploration, perseverance, and the quest for knowledge. As we continue our journey through the landscape of medicine, we carry forward the lessons learned from these early days, striving to honor the legacy of those who sought to conquer the invisible foes that threaten our health. The saga of antibiotics is far from over, and their story continues to unfold in ways that will shape the future of medicine and public health. As the landscape of medicine transformed with the introduction of antibiotics, the scientific community began to explore the profound implications of these discoveries. The mechanisms of action for early antibiotics not only shed light on how they functioned, but also opened avenues for innovative approaches in drug development. Each antibiotic, with its unique method of combating bacterial foes, provided insight into the intricate world of microbiology. Imagine the bacterial cell as a fortress, meticulously designed to guard against intruders. Some antibiotics, like penicillin, target the very walls of this fortress, causing structural failure. Others, such as chloramphenicol, disrupt the assembly lines of protein production, stifling the enemy's ability to mount a defense or launch an attack. Chloramphenicol inhibits the bacterial ribosome, effectively halting protein synthesis. This strategic blockade renders the bacteria unable to grow and reproduce, illustrating the precision with which these early pharmaceuticals could operate. The impact of these advancements extended beyond individual cases of illness. They altered the fabric of public health itself. The early use of antibiotics ushered in a new era of medicine where once deadly infections became treatable. Prior to the advent of these drugs, diseases like scarlet fever and syphilis were often fatal, with little recourse for those afflicted. The onset of antibiotics transformed these grim realities into manageable conditions, paving the way for a more hopeful outlook on healthcare. Consider the story of Howard Florey and Ernst Boris Chain, whose collaborative efforts were instrumental in bringing penicillin from the laboratory to the masses. Driven by the urgency of World War II, they worked tirelessly to mass-produce the antibiotic, recognizing its potential to save countless lives on the battlefield. Florey once remarked, "The great thing about penicillin is that it has no side effects." While this statement reflects the enthusiasm of the time, it also underscores the excitement surrounding the potential of antibiotics to alleviate suffering. As antibiotics gained prominence, the medical community began to appreciate their role in surgical procedures. Before their widespread use, surgery was a risky endeavor, fraught with the threat of infections that could complicate or even nullify the benefits of the operation. With antibiotics in hand, surgeons could proceed with greater confidence, knowing that they had the means to combat post-operative infections. This shift enabled advancements in surgical techniques and opened the door to complex procedures that were previously deemed too perilous. Yet the triumph of antibiotics was accompanied by an unexpected challenge: the emergence of antibiotic resistance. As use proliferated, so too did the evolution of bacteria. Imagine the relentless struggle for survival in the microbial world, where those bacteria that could adapt to resist the effects of antibiotics thrived, while others succumbed. The rapid development of resistance highlighted a crucial aspect of antibiotic use: the need for stewardship and responsible prescribing practices. This phenomenon serves as a reminder that, while antibiotics offer remarkable benefits, they must be used judiciously to maintain their efficacy. The ongoing battle against resistant strains of bacteria has spurred a renewed focus on research and innovation. Scientists are now exploring alternatives to traditional antibiotics, seeking new ways to outsmart these cunning pathogens. From bacteriophage therapy, which harnesses viruses that specifically target bacteria, to the potential of antimicrobial peptides derived from the innate immune systems of various organisms, the quest for solutions continues. Each exploration reflects a commitment to understanding the complexities of microbial life and the challenges it presents. As we navigate this intricate landscape, we must also consider the role of education in combating antibiotic resistance. Public awareness of the appropriate use of antibiotics is vital. How often do we hear about the importance of completing a prescribed course of antibiotics, even when symptoms improve? This practice not only ensures that the infection is fully eradicated, but also helps prevent the development of resistant bacteria. Empowering individuals with knowledge about these medications can significantly influence public health outcomes. The journey of antibiotics from their serendipitous discovery to their early applications illustrates the interplay between science and society. Each breakthrough is not just a testament to human ingenuity, but also a reflection of our collective responsibility to wield such power wisely. As we forge ahead, we must be mindful of the lessons learned from the past, striving to balance the incredible potential of antibiotics against the need for sustainable practices. In this ever-evolving narrative, the future of antibiotics hangs in the balance. What new discoveries await us? Will we find innovative approaches that circumvent the challenges of resistance? The answers remain elusive, yet the pursuit of knowledge continues unabated. The story of antibiotics is a reminder that science is a journey, one filled with twists and turns, breakthroughs and setbacks, each contributing to a broader understanding of life itself. As we ponder the impact of these remarkable discoveries, we are left with a sense of wonder at the intricate world of bacteria, antibiotics, and human health. Each chapter of this ongoing saga invites us to reflect on our relationship with these powerful tools and the responsibility that comes with their use. The journey is far from over, and as we continue to explore the legacy of antibiotics, we do so with an eye toward a future that holds the promise of even greater advancements in medicine. Chapter 3. The Mechanisms of Resistance: How Bacteria Adapt. In the intricate world of bacteria, the mechanisms that allow these microorganisms to adapt and thrive are nothing short of remarkable. At the heart of this adaptability lies the intricate process of genetic mutation and horizontal gene transfer. Imagine a vast library of genetic information, where each bacterium holds a unique book filled with instructions for survival. The pages of this book can be rewritten, edited, or even shared with others, enabling bacteria to rapidly adjust to their ever-changing environments. Genetic mutation occurs naturally and is a fundamental aspect of bacterial life. Picture a single bacterium quietly replicating its DNA as it divides. During this process, occasional errors may creep in, introducing changes to the genetic code. These mutations can be harmless, or they may result in new traits that enhance survival. In the presence of antibiotics, for instance, a random mutation might confer resistance, allowing that bacterium to withstand the very treatment designed to eliminate it. The beauty of this process lies in its simplicity yet profound impact. Small changes can lead to significant advantages. Now, let us explore the fascinating phenomenon of horizontal gene transfer. Unlike genetic mutation, which occurs within a single organism, horizontal gene transfer allows bacteria to exchange genetic material with one another, regardless of their lineage. This transfer can happen in several ways: transformation, transduction, and conjugation. Imagine a bustling marketplace where ideas and resources are shared freely among all participants. In this context, bacteria can acquire advantageous genes from their neighbors, making them more resilient to threats. Transformation involves the uptake of naked DNA from the environment. Picture a bacterium wandering through a landscape strewn with fragments of genetic material left behind by its deceased counterparts. When it encounters this DNA, it can absorb it, incorporating new traits into its own genetic repertoire. Transduction, on the other hand, occurs with the help of viruses that infect bacteria, known as bacteriophages. These phages can inadvertently carry genetic material from one bacterium to another, transferring resistance genes like a courier delivering a vital message. Finally, conjugation involves direct contact between two bacteria, where one bacterium transfers a segment of DNA to another through a specialized structure known as a pilus. This method enables rapid sharing of survival traits, including those linked to antibiotic resistance. As we reflect on the implications of these processes, consider the words of the renowned microbiologist Joshua Lederberg, who said, bacteria are the most successful organisms on Earth. Their ability to adapt and evolve through genetic mutation and horizontal gene transfer underscores this statement. They are not merely surviving, they are thriving and overcoming challenges that would be insurmountable for many other forms of life. The mechanisms of antibiotic resistance in bacteria are varied and sophisticated. Some bacteria may produce enzymes that break down antibiotics, rendering them ineffective. Others may alter their cellular structures to prevent antibiotics from entering. Imagine these bacteria as skilled artisans, crafting barriers and defenses that thwart the very agents meant to destroy them. One common example is the production of beta-lactamase enzymes, which can deactivate penicillin and its derivatives. This ability to modify their surroundings allows bacteria to persist in the face of medical interventions. Another fascinating mechanism is the alteration of drug targets. In this scenario, bacteria can change the very structures within their own cells that antibiotics aim to disrupt. It is much like a fortress that changes its gates and walls, making it impenetrable despite ongoing attacks. By modifying their target sites, these bacteria can evade the effects of antibiotics, ensuring their survival and proliferation. Additionally, some bacteria may actively pump out antibiotics before they can exert their effects, using specialized efflux pumps. Picture these pumps as vigilant sentinels, constantly on guard, expelling intruders before they can cause harm. This active defense mechanism highlights the lengths to which bacteria will go to protect themselves from therapeutic agents. A crucial player in the spread of antibiotic resistance is the plasmid, a small circular piece of DNA that exists independently of the bacterial chromosome. These plasmids can carry genes that confer resistance to antibiotics and can easily be transferred between bacteria through horizontal gene transfer. Imagine plasmids as tiny treasure chests, each containing valuable information that can dramatically change the fortunes of a bacterium. When one bacterium acquires a plasmid with resistance traits, it becomes a beacon of survival in a hostile environment. The significance of plasmids extends beyond individual bacteria. They can facilitate the rapid spread of resistance traits across entire populations. In a world where antibiotics are frequently used, and sometimes misused, these plasmids act as conduits for resistance, allowing bacteria to share survival strategies with astonishing speed. The result is a complex web of resistance that poses a significant challenge to modern medicine. As we consider the implications of these mechanisms, it is essential to acknowledge the contributions of visionary scientists who have advanced our understanding of bacterial resistance. One such figure, Paul Ehrlich, famously aspired to find a magic bullet that would target pathogens without harming the host. While the discovery of antibiotics initially fulfilled this dream, the emergence of resistance has revealed the limitations of such approaches. The evolution of bacteria is a reminder of nature's unpredictable and relentless course. In the realm of antibiotic resistance, the challenges we face are vast. The World Health Organization has identified antibiotic resistance as a global health crisis, warning that we may soon enter a post-antibiotic era where common infections become untreatable. This urgency compels us to reevaluate our relationship with these powerful medications and to seek innovative solutions that can outpace bacterial evolution. As we imagine the future, we can draw hope from the resilience of scientific inquiry. Researchers are exploring novel strategies, including the development of new antibiotics, phage therapy, and even the use of bacteriocins, which are proteins produced by bacteria that can inhibit the growth of similar or closely related bacterial strains. Each of these approaches represents a step toward reclaiming control in the ongoing battle against resistant bacteria. In this journey, one cannot help but reflect on the adaptability of life itself. Bacteria, in their microscopic world, hold lessons on survival that reach far beyond their tiny forms. They remind us that resilience is woven into the very fabric of existence, a testament to the power of change and adaptation. As we continue to uncover the mechanisms of resistance, we gain insight into not only the challenges we face, but also the remarkable ingenuity that nature has to offer. The exploration of bacteria is an endless voyage, one that continues to reveal the mysteries of life and the intricate relationships that bind all living beings. In this intricate landscape of bacterial resistance, it is essential to recognize the role of plasmids as agents of change, not merely as passive carriers of genetic information. These small, circular DNA molecules are akin to a mobile library, offering a treasure trove of genes that can confer new abilities upon bacteria. Imagine a bustling city where ideas flow freely and new innovations are shared among the residents. In this scenario, plasmids act as messengers, allowing bacteria to acquire resistance traits that enhance their survival prospects in a world increasingly dominated by antibiotics. The journey of plasmids begins with their ability to replicate independently within a bacterial cell. This autonomous nature allows them to multiply rapidly, ensuring that their valuable cargo can be passed on to subsequent generations. When a bacterium encounters stressors, such as antibiotics, the presence of a plasmid with resistance genes can mean the difference between life and death. Picture a community of bacteria, some equipped with plasmids, while others remain vulnerable. As antibiotic pressure mounts, those with plasmids thrive, rapidly outnumbering their unprotected counterparts. The adaptability of plasmids extends beyond individual bacteria. Through horizontal gene transfer, they can move between different species, facilitating a rapid exchange of traits. Imagine a network of interconnected pathways where information and genetic material flow seamlessly between diverse bacterial populations. This phenomenon not only accelerates the spread of resistance, but also contributes to the emergence of multi-drug resistant strains, presenting a formidable challenge to medical science. Consider the work of scientists like Michael Altman, who has studied the dynamics of plasmid transfer and its implications for public health. Altman emphasizes that understanding these mechanisms is crucial for developing strategies to combat antibiotic resistance. He notes that the evolutionary arms race between bacteria and antibiotics is a complex and ever-evolving battle. This battle underscores the need for vigilance and innovation as we seek to outsmart these resilient organisms. As we delve deeper into the specific mechanisms of antibiotic resistance, we encounter a fascinating array of adaptations. Some bacteria employ a strategy known as modification of the antibiotic target, where they alter the very sites that antibiotics are designed to attack. Imagine a castle under siege, where the defenders change the layout of the walls to thwart the invaders. In this case, the bacteria modify their ribosomes or cell wall components, making it difficult for antibiotics to bind effectively and disrupt their functions. Another prominent mechanism involves the production of efflux pumps, which serve as cellular bouncers, ejecting antibiotics before they can accumulate and exert their harmful effects. Picture these pumps as vigilant guards at the entrance to a nightclub, ensuring that only the right guests are allowed in, while unwanted intruders are swiftly removed. Efflux pumps are particularly adept at removing a wide range of antibiotics, demonstrating the bacteria's remarkable capacity to adapt to various threats. The emergence of plasmid-borne resistance genes further complicates the landscape. Not only do these plasmids facilitate the transfer of resistance traits, but they can also carry multiple genes, allowing bacteria to resist multiple classes of antibiotics simultaneously. Imagine a toolkit filled with an array of tools, each designed to tackle different problems. When bacteria acquire such plasmids, they become equipped to face a diverse range of challenges, thereby enhancing their survival in hostile environments. In this era of antibiotic resistance, the urgency for new solutions has never been greater. Scientists are actively seeking ways to disrupt the mechanisms that underpin bacterial resilience. Innovative strategies are being explored, such as the development of inhibitors that target efflux pumps, or the design of antibiotics that can bypass the modified targets. These efforts reflect a commitment to reclaiming the upper hand in our ongoing struggle against resistant bacteria. As we ponder the future, it is fascinating to consider the potential of alternatives to traditional antibiotics. Phage therapy, for example, harnesses the power of bacteriophages, viruses that specifically infect bacteria to target and kill resistant strains. Imagine a precision-guided missile homing in on its target with remarkable accuracy. This approach offers a glimmer of hope, particularly in cases where conventional antibiotics have failed. Moreover, the exploration of bacteriocins, proteins naturally produced by bacteria that inhibit the growth of similar or closely related strains, represents another exciting frontier. These naturally occurring substances may hold the key to developing new treatments that can effectively combat resistant bacteria while preserving the delicate balance of the microbiome. As we navigate this multifaceted landscape, the resilience and adaptability of bacteria serve as a profound reminder of nature's ingenuity. Their ability to withstand the pressures of antibiotics is a testament to their evolutionary prowess. The journey to understand and combat antibiotic resistance is one that calls for collaboration, creativity, and a relentless pursuit of knowledge. In this ongoing quest, we find ourselves at a crossroads, where the lessons learned from the microbial world can inform our strategies moving forward. The interplay between bacteria, their mechanisms of resistance, and the human response to these challenges is a dynamic and evolving narrative, one that will continue to unfold as we seek to maintain the efficacy of antibiotics in the face of relentless adaptation. The exploration of bacterial resistance is not merely a scientific endeavor. It is a reflection of the broader struggle for survival in an ever-changing world. As we deepen our understanding of these mechanisms, we not only confront a pressing public health crisis, but also uncover the intricate connections that bind all living organisms together in this remarkable tapestry of life. Chapter 4. Natural Selection: Driving Forces Behind Resistance. In the vast and intricate world of microbial life, natural selection serves as a powerful force driving the evolution of bacteria. Imagine a landscape teeming with microscopic organisms, each vying for survival in an environment filled with challenges. In this realm, the principles of natural selection play out with remarkable clarity. When we consider the sheer adaptability of bacteria, it becomes evident that their survival often hinges on a series of selective pressures. These pressures can be likened to a relentless tide, shaping the characteristics of bacterial populations over time. Natural selection operates on the principle that individuals within a population exhibit variations in traits, some of which may confer advantages when faced with specific environmental pressures. Picture a colony of bacteria exposed to an antibiotic. Among them, a few may possess a genetic mutation that allows them to resist the drug's effects. These resilient outliers, having evaded the lethal impact of the antibiotic, will thrive and reproduce, passing on their resistant traits to future generations. This cycle unfolds repeatedly, creating a dynamic interplay between survival and adaptation. As we explore the environmental pressures that promote the development of resistance, we can begin to understand the profound impact of human activity on microbial evolution. The widespread use of antibiotics, for instance, has dramatically altered the landscape in which bacteria exist. Imagine a once diverse community of bacteria facing a sudden influx of antibiotics, akin to a storm sweeping through a forest. The sensitive species are quickly diminished, while those with inherent resistance are left to flourish. This selective environment encourages the proliferation of resistant strains, effectively steering the course of bacterial evolution. One telling example can be found in the emergence of methicillin-resistant Staphylococcus aureus, commonly known as MRSA. Initially, methicillin was heralded as a breakthrough in the fight against staphylococcal infections, offering a potent weapon against a common and dangerous pathogen. However, the extensive use of this antibiotic in both healthcare and agricultural settings created a fertile ground for resistance to develop. The bacteria that survived exposure to methicillin were those that harbored mutations or acquired resistance genes, enabling them to adapt to this new challenge. The rise of MRSA serves as a cautionary tale, reminding us of the delicate balance between our efforts to combat bacterial infections and the unintended consequences that can arise. Another captivating case study is that of Mycobacterium tuberculosis, the bacterium responsible for tuberculosis. This resilient organism has exhibited an extraordinary ability to evolve in response to treatment pressures. The use of antibiotics, particularly in incomplete or inappropriate courses, has led to the emergence of multi-drug resistant strains. Imagine a city besieged by a relentless enemy, where only the most cunning and resourceful defenders remain. Similarly, M. tuberculosis has adapted by developing mechanisms to withstand multiple antibiotics, complicating treatment efforts, and posing significant public health challenges. The story of bacterial resistance is not merely a tale of survival. It is also one of innovation and adaptation. Bacteria possess remarkable genetic flexibility, allowing them to respond to environmental changes with astounding speed. The process of horizontal gene transfer, which we touched upon in our previous exploration of bacterial mechanisms, plays a crucial role in this narrative. Through this process, bacteria can share resistance traits with one another, akin to passing secret knowledge among allies in the face of adversity. This genetic exchange can occur through various means, including transformation, transduction, and conjugation, facilitating a rapid spread of resistance genes within communities. Consider the implications of this sharing. Within a population of bacteria, a single resistant strain can pass its advantageous traits to neighboring strains, creating a wave of resistance that can sweep through an entire community. This interconnectedness highlights the urgency of addressing antibiotic misuse and overprescription, as each decision can ripple through the microbial world, influencing the trajectory of resistance. In addition to human influence, environmental factors such as sanitation practices, agricultural methods, and even climate change contribute to the emergence of resistant strains. Bacteria are not isolated entities. They exist within complex ecosystems, interacting with a multitude of organisms and environmental factors. The introduction of antibiotics into agricultural practices, for instance, has led to increased resistance among bacterial populations in livestock and crops. The consequences of these practices extend beyond agricultural fields, as resistant bacteria find their way into the human population, further complicating efforts to manage infections. A poignant example of this phenomenon can be seen in the case of Escherichia coli, a bacterium commonly found in the intestines of humans and animals. Certain strains of E. coli have developed a resistance to multiple antibiotics, often originating from agricultural runoff that introduces resistant bacteria into the environment. This scenario underscores the interconnectedness of human health and environmental practices, illustrating how the choices we make can have far-reaching effects on microbial populations. As we reflect on the driving forces behind resistance, it is essential to recognize the role of research and innovation in combating this complex issue. Scientists are continually exploring new avenues to understand and mitigate the emergence of resistant strains. The development of novel antibiotics, alternative treatments, and strategies to enhance the efficacy of existing drugs is an ongoing pursuit. Each breakthrough offers a glimmer of hope in the battle against bacterial resistance, yet it also emphasizes the need for a proactive approach to antibiotic stewardship. Imagine a future where we cultivate an environment that discourages the emergence of resistance, where responsible antibiotic use is the norm rather than the exception. This vision requires collaboration among healthcare providers, researchers, policymakers, and the public to foster a comprehensive understanding of the consequences of our actions. Education plays a vital role in this effort, empowering individuals to make informed choices about antibiotic use and encouraging a sense of responsibility towards microbial health. The story of natural selection and resistance is one of resilience, adaptation, and the intricate dance of life at the microscopic level. As bacterial populations evolve in response to the pressures we impose, they remind us of the delicate balance that underpins our existence. Each resistant strain that emerges serves as a testament to the tenacity of life, a call to action for all of us to engage thoughtfully in the stewardship of our health and the environment. In the face of these challenges, we must remain vigilant and committed to understanding the delicate interplay of forces that shape the microbial world. The journey ahead is filled with uncertainties, yet it also brims with possibilities for innovation and collaboration. Through a concerted effort, we can strive to mitigate the effects of resistance, fostering a healthier relationship with the bacteria that share our world. As we continue to explore this intricate landscape, let us do so with a sense of wonder and responsibility, recognizing the profound impact our choices can have on the microbial tapestry that underpins life itself. In the ongoing saga of microbial evolution, the principles of natural selection reveal a fascinating narrative woven through the lives of countless bacterial species. This narrative is not solely about survival. It embodies the intricate relationship between bacteria and their environments, shaped by both natural phenomena and human actions. As we explore this relationship further, it becomes clear that certain environmental pressures catalyze the development of resistance, pushing bacteria to adapt in ways that can be both astonishing and alarming. Consider the concept of environmental pressures. These pressures arise from various sources, such as changes in temperature, nutrient availability, or the presence of toxic substances. Each of these factors can influence bacterial survival. For instance, imagine the impact of industrial pollution on local microbial populations. Heavy metals and chemical waste can create a hostile landscape for many organisms, but for some bacteria, these same challenges can serve as catalysts for innovation. Those capable of developing resistance to these pollutants can thrive in environments where others perish, gaining a competitive edge. In the agricultural realm, the introduction of herbicides and pesticides has similarly stimulated resistance development among bacterial populations. The widespread application of these chemicals serves as a powerful selective force. Picture a field where certain bacterial strains are exposed to a potent herbicide. Over time, those bacteria that manage to survive will be the ones that possess the necessary genetic adaptations. This selective pressure creates a scenario where resistant strains not only endure, but also proliferate, potentially leading to a cascade of resistance that affects not just the targeted pests, but also beneficial microbial communities essential for soil health. One striking case study that exemplifies this phenomenon is that of Pseudomonas aeruginosa, a bacterium notorious for its resilience in a variety of environments, including soil, water, and even human hosts. P. aeruginosa is particularly adept at developing resistance to antibiotics and disinfectants, a trait that has made it a formidable pathogen in healthcare settings. Its ability to thrive in harsh conditions, often fueled by human-induced environmental changes, illustrates how bacterial resistance can emerge as a direct response to selective pressures. This bacterium has been known to adapt its metabolic pathways, allowing it to utilize alternative carbon sources when traditional nutrients are scarce. As a result, it can flourish in environments where many other organisms would struggle to survive. The story of Pseudomonas aeruginosa is not just one of resistance. It is also a testament to the interconnectedness of ecological systems. The rise of resistant strains often has implications that extend beyond the immediate environment. For example, when P. aeruginosa becomes resistant to treatment in a hospital setting, it can lead to severe infections in vulnerable patients, highlighting how the pressures exerted in one context can reverberate throughout the healthcare system. This interconnectedness serves as a reminder of the importance of maintaining a holistic view of microbial health and the environments in which they exist. Furthermore, the emergence of resistance is not confined to bacteria alone. It extends into the realm of viruses and fungi, illustrating a broader narrative of survival and adaptation. In the world of virology, for instance, the influenza virus demonstrates a similar ability to evolve rapidly in response to immunological pressures. This phenomenon should prompt us to ponder, how do these adaptive processes shape our understanding of infectious diseases? The interplay between host immune responses and pathogen evolution reveals a complex battlefield where the stakes are high, affecting public health on a global scale. As we delve deeper into case studies of resistance emergence, we can observe the role of specific bacterial characteristics in shaping these narratives. One such characteristic is biofilm formation, a strategy employed by many bacteria to enhance their survival. Imagine a community of bacteria creating a protective layer, a biofilm, that shields them from environmental threats, including antibiotics. This structure not only fosters a conducive environment for the bacteria within, but also allows them to exchange genetic material more freely, accelerating the spread of resistance traits. The biofilm-forming capabilities of species like Staphylococcus epidermidis, a common inhabitant of human skin, illustrate the potential for resistance development in clinical settings. This bacterium, while often harmless, can become problematic when it forms biofilms on medical devices such as catheters or prosthetic joints. Within these biofilms, the bacteria can evade the immune system and resist antibiotic treatment, leading to persistent infections that are challenging to eradicate. What does this tell us about our approach to infection control and treatment? It suggests that our strategies must evolve in tandem with the bacteria we aim to combat. Emphasizing the importance of preventing biofilm formation and developing novel therapeutic approaches to disrupt these communities can be crucial in managing resistant infections. In light of these insights, the question arises: how can we apply our understanding of natural selection and resistance development to create effective interventions? The answer lies in a multifaceted approach that combines research, education, and responsible practices. Developing new antibiotics is essential, but it is equally important to ensure their judicious use. By fostering awareness of the consequences of antibiotic misuse, we can contribute to a healthier microbial community, one that resists the pressures leading to resistance. As we reflect on the ongoing battle between bacteria and the forces that shape their evolution, it becomes evident that each interaction in this intricate web of life carries profound implications. The principles of natural selection guide the narrative of bacterial resistance, driven by environmental pressures that challenge their very existence. This understanding invites us not only to observe, but also to engage actively in the stewardship of our microbial companions, recognizing that our choices ripple through the delicate balance of life around us. In the end, the story of resistance is not solely one of conflict, but also of resilience and adaptation. Each bacterial strain that emerges in response to environmental pressures serves as a reminder of the tenacity of life and the ongoing dance of evolution that surrounds us. The path forward requires careful navigation through the complexities of microbial life, fostering a future that embraces both innovation and responsibility. Chapter 5: The Role of Overuse, Antibiotics in Medicine and Agriculture. In the intricate web of human health, antibiotics play a crucial and complex role. Their introduction into medicine marked a pivotal shift in our ability to combat bacterial infections, transforming once deadly diseases into manageable conditions. Yet this remarkable tool comes with significant responsibilities. As we explore the patterns of antibiotic use in both human medicine and agriculture, we uncover a narrative that intertwines innovation with caution. Imagine a bustling hospital, where doctors and nurses work tirelessly to save lives. In this environment, antibiotics are the first line of defense against infections that can quickly escalate and threaten life. Yet the very act of prescribing these medications requires careful deliberation. Physicians must weigh the benefits of treatment against the potential for resistance. The decision to prescribe an antibiotic is not taken lightly. It reflects a delicate balance between immediate patient needs and long-term implications for public health. In agriculture, the landscape shifts, but the stakes remain high. Farmers utilize antibiotics not only to treat sick animals, but also to promote growth and prevent disease in healthy livestock. Picture vast fields dotted with livestock, where antibiotics are administered to ensure that animals thrive in crowded conditions. This practice, while boosting productivity, introduces antibiotics into the environment in ways that can have far-reaching consequences. The routine use of these medications in farming creates an ecosystem ripe for the development of resistant bacterial strains. As we consider the consequences of overprescribing and misuse, we begin to see the shadows cast by our reliance on antibiotics. When these powerful drugs are used excessively or inappropriately, they can lose their effectiveness, leading to what many health professionals now deem a crisis. The World Health Organization has cautioned that antibiotic resistance is one of the biggest threats to global health today. Reflect on this. What happens when antibiotics become less effective? Infections that were once easily treatable can lead to prolonged illness, higher medical costs, and even increased mortality rates. The very bacteria that we aim to control adapt and evolve, developing mechanisms to withstand the drugs designed to eliminate them. The impact on bacterial populations is profound. Just as we learned about the principles of natural selection in the previous chapter, the overuse of antibiotics creates a selective pressure that favors resistant strains. Imagine a scenario where a population of bacteria is exposed to an antibiotic. In this environment, those with mutations that confer resistance survive and reproduce, passing on their traits to future generations. The result is a burgeoning population of resistant bacteria that can spread not only among individuals, but also through communities, amplifying the threat. Consider the case of a common bacterium, Escherichia coli, usually harmless in the gut. Yet, when subjected to antibiotics, certain strains can evolve into formidable pathogens. These resistant strains can lead to urinary tract infections that are increasingly challenging to treat, resulting in a growing public health concern. Here we see a clear example of how the misuse of antibiotics leads to unforeseen consequences that ripple through our healthcare systems. The story of antibiotics is not solely a tale of science and medicine. It also intertwines with the lives of extraordinary individuals who have sought to address these challenges. Take the work of Dr. Margaret Chan, former Director General of the World Health Organization. She emphasized the importance of responsible antibiotic use, stating, quote, "A world without antibiotics would be a world without modern medicine." Her words resonate as a reminder of how intertwined our health practices are with the stewardship of these vital resources. The concept of stewardship becomes increasingly vital as we acknowledge the relationship between human actions and bacterial evolution. By understanding the patterns of antibiotic use, we can begin to envision strategies to mitigate resistance. Thoughtful prescribing practices, patient education, and the development of new antibiotics and alternative therapies are all part of a multifaceted approach to preserving these life-saving drugs. Imagine a world where antibiotics remain effective, where infections can be treated without the looming threat of resistance. This vision is not just a dream. It is a possibility that requires collective action and awareness. Each prescription, each application in agriculture represents a decision that can either contribute to the problem or help safeguard our future. As we explore the impact of antibiotic overuse on resistance rates, we must consider the broader implications. The agricultural sector plays a significant role in this narrative. The use of antibiotics in livestock not only affects animal health, but also influences human health through the food supply. Resistant bacteria can find their way into meat and other animal products, leading to potential infections in consumers. In this interconnected world, the actions taken in one arena can echo across the globe. The concept of the One Health approach underscores the interconnectedness of human, animal, and environmental health. By recognizing that the health of humans is intrinsically linked to the health of animals and ecosystems, we can forge a path toward more sustainable practices. The consequences of antibiotic overuse extend beyond the individual. They create a community of bacteria that can share resistance genes, further complicating our ability to combat infections. Picture a network of bacteria, each one capable of exchanging genetic material and amplifying resistance traits. This phenomenon underscores the urgency of addressing antibiotic use, not just in medicine, but across all sectors that interact with bacteria. As we reflect on the patterns of antibiotic use, we are confronted with a pressing question. How can we shift our practices to ensure the continued effectiveness of these essential medications? The answer lies in a multifaceted strategy that emphasizes education, regulation, and innovation. By fostering a culture of responsible antibiotic use, we can begin to reestablish the balance necessary to protect both human health and the delicate ecosystems that sustain us. In considering the impact of our choices, we are reminded of the words of Sir Alexander Fleming, who cautioned, "The greatest possibility of evil is in the misuse of antibiotics." His insight highlights the profound responsibility that accompanies the discovery and use of these powerful drugs. As stewards of health, it is incumbent upon us to tread carefully, ensuring that our actions today do not jeopardize the well-being of future generations. As we draw our attention back to the bacterial populations that surround us, we witness the remarkable adaptability of these organisms. They are not merely passive recipients of human action. They respond and evolve in ways that challenge our medical practices. This dynamic relationship underscores the need for ongoing research and vigilance in our approach to antibiotics. In the face of antibiotic resistance, the call for innovation becomes ever more pressing. Scientists around the world are exploring novel approaches to combat bacterial infections, from bacteriophage therapy to the development of new classes of antibiotics. Imagine a future where we harness the power of nature itself to counteract the threats posed by resistant bacteria. As we navigate this critical juncture in healthcare, the role of overuse becomes clear. It is a reminder that the choices we make, both as individuals and as a society, have profound implications. By fostering a deeper understanding of antibiotics and their impact on health, we can cultivate a more sustainable relationship with these essential tools. The journey toward addressing antibiotic resistance is undoubtedly complex, yet it is a journey we must embark upon with determination and foresight. As we continue to explore this vital topic, let us remain mindful of the delicate balance between innovation and caution, always striving to protect the health of both ourselves and the world around us. As we further explore the consequences of overprescribing antibiotics, it becomes increasingly clear that the implications extend beyond mere effectiveness. The fabric of our health systems is intricately woven with various threads, each representing a different facet of antibiotic use, from human medicine to agricultural practices. What happens when these threads fray and unravel? The potential for a public health crisis looms large on the horizon. In the realm of agriculture, the integration of antibiotics into animal husbandry has created a complex scenario. Farmers often administer these drugs not solely for treating illness, but to enhance growth and prevent disease in otherwise healthy animals. Consider the vast poultry farms where thousands of birds are raised in close quarters. The use of antibiotics in these settings is often routine, contributing to impressive productivity rates. However, this method comes at a price. The proliferation of resistant bacteria in livestock can infiltrate the food chain, leading to human exposure through consumption of animal products. Imagine the journey of a resistant bacterium. It begins within the gut of a livestock animal, thriving on the antibiotics intended for its growth. When this animal is processed for food, the bacteria can survive cooking and find their way onto our plates. The result? Individuals consuming these products may unknowingly ingest resistant strains, leading to infections that are difficult, if not impossible, to treat. The interconnectedness of our food supply and antibiotic use becomes a clear illustration of how one practice can ripple across ecosystems, impacting human health in unforeseen ways. As we delve deeper into the impact of antibiotic overuse on resistance rates, we encounter a chilling statistic: the emergence of multi-drug-resistant bacteria. These formidable pathogens not only resist one class of antibiotics, but can withstand multiple types, rendering standard treatments ineffective. The implications for surgical procedures, cancer treatments, and even routine infections are staggering. Imagine a world where a simple surgery becomes a high-stakes gamble, due to the risk of post-operative infections that cannot be successfully treated. This scenario is not a distant future but a reality that is already unfolding. The emergence of strains such as methicillin-resistant Staphylococcus aureus serves as a dire warning. This bacterium, once easily treatable, has evolved into a scourge of hospitals and communities alike. The story of MRSA is a testament to the resilience of bacteria and the unintended consequences of human actions. As we reflect on these developments, we are led to a pivotal question. How do we turn the tide against antibiotic resistance? The answer lies in a multi-pronged approach that encompasses education, regulation, and innovation. Health professionals and agricultural experts alike must champion responsible antibiotic use, promoting practices that minimize unnecessary prescriptions. Education plays a vital role in this endeavor. Imagine patients being empowered with knowledge about their health, understanding when antibiotics are truly necessary and when they are not. This shift in awareness could lead to a dramatic reduction in the misuse of these medications. Similarly, farmers can be educated about alternatives to antibiotics, such as improved animal husbandry practices that focus on hygiene and nutrition. Regulation also holds a key position in combating antibiotic overuse. Policies that limit the use of antibiotics in livestock, ensuring that they are reserved for therapeutic purposes rather than growth promotion, are crucial to curbing the spread of resistance. Countries that have implemented such measures have witnessed a positive impact on both animal health and public health outcomes. Innovation must accompany these efforts. Scientists are tirelessly exploring new avenues for treatment, ranging from bacteriage therapies to novel antimicrobial agents. Imagine a future where, instead of relying solely on traditional antibiotics, we harness the natural predators of bacteria to combat infections. This approach not only preserves existing antibiotics, but also reintroduces a layer of complexity in managing bacterial populations. The history of antibiotic development is also rich with stories of extraordinary individuals who have paved the way for change. Dr. Tu Youyou, a Chinese scientist, discovered artemisinin, a groundbreaking treatment for malaria. Her journey demonstrates that perseverance and innovation can lead to breakthroughs, even in the face of daunting challenges. As we face the threat of antibiotic resistance, it is essential to channel that spirit of discovery into finding new solutions. As we consider the ramifications of our antibiotic use, we must also be aware of the ethical responsibilities that accompany our choices. The overuse of antibiotics presents a moral dilemma, raising questions about our duty to protect not only our own health, but the health of future generations. Reflect on the interconnectedness of life. Each decision we make ripples outward, influencing the delicate balance of ecosystems. As we navigate this landscape of antibiotic use and resistance, it is crucial to foster a culture of stewardship that embraces both innovation and responsibility. This journey will require collaboration among scientists, healthcare providers, policymakers, and the public. Imagine a world where we have not only preserved the effectiveness of antibiotics, but have also cultivated a deeper understanding of their role in our lives. The road ahead is fraught with challenges, yet it is also filled with possibilities. As we continue to explore the intricate dynamics of antibiotics, let us remain vigilant stewards of these powerful tools, ensuring that they remain effective for generations to come. The choices we make today will shape the future of medicine, agriculture, and public health, guiding us toward a horizon where antibiotic resistance is no longer a looming threat but a challenge we have collectively overcome. Chapter 6. Global Spread: Tracking Antibiotic-Resistant Bacteria. The journey of antibiotic-resistant bacteria across the globe can be likened to a silent storm, sweeping through communities and countries, often unnoticed until the damage has been done. Understanding how these resistant strains spread is essential for grasping the broader implications for global health. Bacteria possess an incredible ability to thrive in diverse environments, and this adaptability is amplified through various mechanisms of transmission. Consider the ways in which these resilient bacteria travel. They can hitch a ride on the hands of healthcare workers, slip through the cracks of sanitary protocols, or even linger on surfaces in environments like hospitals and clinics. Imagine entering a bustling hospital, where the air is filled with the beeping of machines and the soft murmur of nurses attending to patients. Each surface, doorknobs, countertops, medical equipment, can act as a conduit for bacteria, facilitating the transmission of resistance genes from one organism to another. In addition to person-to-person contact, resistant bacteria can spread through the food supply, particularly in agricultural settings where antibiotic use is prevalent. Picture sprawling farms, where livestock are treated not only for illness, but also routinely administered antibiotics to enhance growth. When these animals are processed for food, they can carry resistant bacteria, which may then find their way into our meals. As we consume these foods, we unknowingly invite potential threats into our own bodies. This cycle raises important questions about food safety and the responsibility we hold in ensuring the health of our communities. Water sources, too, can serve as vectors for antibiotic resistance. Wastewater from hospitals and agricultural runoff can introduce resistant bacteria into rivers, lakes, and even drinking water supplies. Imagine a serene river flowing through a countryside, its crystal waters reflecting the sunlight. Yet beneath this tranquil surface, there can be a hidden risk, bacteria that have adapted to survive, not only in their original environments, but have also acquired new traits that allow them to resist treatment. The implications of antibiotic resistance extend far beyond individual health concerns. They ripple across the fabric of global health. When resistance spreads unchecked, the effectiveness of antibiotics diminishes, leading to longer hospital stays, higher medical costs, and increased mortality. The World Health Organization has warned that we are entering a post-antibiotic era, where simple infections could once again become life-threatening. Global health systems are beginning to feel the weight of this crisis. A hospital that once boasted the ability to treat infections with ease may now find itself grappling with cases that do not respond to standard treatments. This situation is not confined to any one nation. It is a global challenge, requiring collective action. Countries are starting to implement surveillance systems to track the spread of resistant bacteria, aiming to identify outbreaks before they escalate. Outbreaks of antibiotic-resistant bacteria serve as stark reminders of the urgent need for robust management strategies. One notable case occurred in a large urban hospital where a cluster of infections caused by a strain of Klebsiella pneumoniae, resistant to nearly all available antibiotics, emerged. In this instance, healthcare providers faced a daunting challenge. The initial response involved rigorous infection control measures, including enhanced cleaning protocols and the isolation of affected patients. As the outbreak unfolded, it became clear that swift action was paramount. The hospital called upon experts in infectious diseases to assist in managing the crisis. These professionals emphasized the importance of collaboration between departments within the hospital and with public health authorities. In a scenario where time was of the essence, the efforts to contain the outbreak required a coordinated approach, underscoring the interconnectedness of healthcare systems. Another example of such efforts can be found in the management of an outbreak of methicillin-resistant Staphylococcus aureus, or MRSA, in a community setting. This strain of bacteria, notorious for its resistance, rapidly spread among a group of individuals who frequented the same gym. The response involved not only the treatment of those infected, but also educational initiatives aimed at preventing further transmission. Health officials distributed information on hygiene practices and the importance of seeking medical attention for persistent infections. These case studies illuminate the vital role of rapid response and public awareness in managing outbreaks. The stories of those affected remind us of the human element behind the statistics, highlighting the serious consequences of antibiotic resistance on individuals and families. As we reflect on the myriad ways antibiotic-resistant bacteria can spread, we must also consider the responsibility we share in combating this global health threat. The practices we adopt in our daily lives, from how we use antibiotics to our choices about food, play a crucial role in shaping the future of public health. Imagine a world where antibiotics remain effective, where infections can be treated without the looming threat of resistance. This vision is not unattainable, but it requires a collective commitment to responsible antibiotic use and ongoing research into alternative treatments. As we advance into this new frontier, let us hold on to the hope that through collaboration, education, and innovation, we can reclaim the narrative surrounding antibiotic resistance and ensure a healthier future for all. The challenge ahead is formidable, yet within it lies the potential for remarkable breakthroughs. Scientists and healthcare professionals worldwide are working tirelessly to develop new strategies and therapies to combat resistant bacteria. Through understanding the mechanisms of resistance and the pathways of transmission, we can build a framework for effective prevention and control measures. As we embrace the complexity of this issue, we are reminded of the importance of vigilance and adaptability. Just as bacteria have evolved to survive, we too must evolve in our approaches to healthcare and disease prevention. Together, we can foster a culture of awareness and responsibility, one that prioritizes the health of our communities and the integrity of our medical systems. In this ongoing journey, each of us plays a part. The choices we make, the conversations we engage in, and the actions we take can contribute to a more sustainable and resilient future. The landscape may be challenging, but it is also filled with opportunities for growth and innovation. Through concerted efforts, we can navigate the complexities of antibiotic resistance and work towards a world where these vital medications continue to save lives. As we move forward, let us remain steadfast in our commitment to understanding and addressing the global spread of antibiotic-resistant bacteria. The stakes are high, but so too is our capacity for change. With every step we take, we draw closer to a future where the threat of resistance is diminished, and the promise of effective treatment remains a reality for generations to come. The spread of antibiotic-resistant bacteria is not merely a concern confined to hospitals or farms. It is a pervasive threat that has begun to infiltrate the very fabric of our global health systems. As we reflect on the mechanisms of transmission, it becomes apparent that these resilient microorganisms can travel across borders with remarkable ease. Consider how international travel has transformed our world into a more interconnected place. Individuals move from one country to another, often carrying with them not just their belongings but also the bacteria that reside within them. In the bustling airports and crowded airplanes, a single cough or sneeze can potentially disperse these resistant strains, igniting outbreaks in places that may have previously been unaffected. Furthermore, the global trade of goods, particularly food, serves as another conduit for these bacteria. Imagine a shipment of fresh produce traveling thousands of miles before reaching your local grocery store. If any step in the supply chain involves contaminated water or soil, the risk of introducing resistant bacteria into new territories escalates. The intricate web of our global economy, while beneficial, inadvertently facilitates the spread of these pathogens, turning local health issues into international crises. The implications of antibiotic resistance extend beyond the immediate health of individuals. They present significant challenges for public health systems worldwide. Countries with limited resources may find themselves particularly vulnerable, as the escalating costs associated with treating resistant infections strain already fragile healthcare infrastructures. The World Health Organization has emphasized that without effective interventions, the very foundations of modern medicine, such as surgical procedures and chemotherapy, could be compromised, as infections that were once easily treatable become increasingly difficult to manage. Take, for instance, the story of a rural clinic in a developing nation facing an outbreak of resistant bacteria. The staff, already stretched thin, find themselves grappling with patients who present with infections that do not respond to conventional antibiotics. The clinic's limited access to advanced medical technologies and treatment options further complicates their ability to respond effectively. This scenario underscores the urgent need for global solidarity, as the fight against antibiotic resistance cannot be confined to individual nations. Collaborative efforts are essential to strengthen healthcare systems, improve surveillance, and establish guidelines for the responsible use of antibiotics. Examining specific case studies reveals the real-world consequences of this crisis. One notable incident occurred in a densely populated city, where a sudden rise in cases of carbapenem-resistant Enterobacteriaceae sent alarm bells ringing throughout the healthcare community. The initial response involved rigorous testing to identify the source of the outbreak. Local health authorities worked tirelessly, tracing connections between patients and uncovering a common thread: an unregulated clinic that had been administering antibiotics without appropriate medical oversight. In a race against time, public health officials rallied to implement containment measures. Educational campaigns were launched, aiming to inform the population about the dangers of self-medication and the importance of seeking proper medical guidance. The outbreak served as a wake-up call, illuminating the need for stringent regulations in healthcare practices, particularly in areas where access to antibiotics may be mismanaged. Another compelling case study emerged from a university campus, where an outbreak of antibiotic-resistant gonorrhea highlighted the complexities of resistance in sexually transmitted infections. Rapid testing revealed that the strain was resistant to multiple first-line treatments. In response, health authorities initiated a comprehensive strategy that combined immediate treatment for affected individuals with outreach efforts to educate students about safe practices. The experience reinforced the notion that education and awareness are vital tools in combating the spread of resistant bacteria, particularly among populations that may be unaware of the risks. These examples illustrate that outbreaks of antibiotic-resistant bacteria are not isolated incidents. They are interconnected events that require a multifaceted approach to management. The stories of those affected remind us that behind each statistic is a person or a family grappling with the consequences of antibiotic resistance. The human experience is woven into the fabric of this crisis, emphasizing the need for compassion and understanding as we navigate these challenging waters. Moreover, as we contemplate the future, it is essential to consider the innovative solutions being explored to combat antibiotic resistance. Researchers and scientists are investigating alternatives such as bacteriophages, viruses that specifically target bacteria. Picture these phages as tiny scouts, seeking out bacteria to destroy while leaving healthy cells unharmed. This burgeoning field of research offers hope, suggesting that we may find new methods to combat infections without further exacerbating resistance. In addition, the development of vaccines targeting common bacterial infections could play a pivotal role in prevention. These vaccines would serve as a shield, protecting individuals from infections before they even occur, thereby reducing the reliance on antibiotics altogether. Imagine a future where routine vaccinations not only safeguard against viral diseases, but also bolster our defenses against bacterial threats. As we immerse ourselves in the narrative of antibiotic resistance, we must also acknowledge the importance of stewardship. Each individual plays a vital role in this collective effort, understanding that responsible antibiotic use is not just a medical concern, but a moral one. The choices made in our homes and communities resonate far beyond our immediate surroundings, impacting the health of our global family. We are at a crossroads, where the decisions we make today can define the trajectory of public health for generations to come. The path forward is illuminated by the possibilities of innovation, collaboration, and education. By embracing these principles, we can foster a future where the legacy of antibiotic resistance is transformed from one of despair to one of hope and resilience. As we continue on this journey, let us remain vigilant and proactive, acknowledging the challenges that lie ahead while also celebrating the strides made in the pursuit of effective solutions. The fight against antibiotic-resistant bacteria is not merely a battle. It is a shared commitment to safeguarding health for ourselves and the generations that will follow. In this endeavor, we find not just our responsibility, but the promise of a healthier, more sustainable world. Chapter 7. Innovations in Treatment, Combating Resistance. In the realm of medicine, the evolution of antibiotic treatments has entered a new chapter, one marked by innovation and adaptability in response to the ever-looming threat of bacterial resistance. Traditional methods, while transformative, face challenges that demand fresh perspectives and new strategies. Imagine a world where the very tools designed to combat infection are rendered ineffective. This is the reality we confront as bacteria evolve, necessitating a shift towards novel approaches in antibiotic development. One promising avenue is the exploration of new compounds derived from natural sources. Scientists are actively seeking out previously overlooked organisms that may harbor unique antibiotic properties. Consider the vast biodiversity found in remote ecosystems, lush rainforests, deep-sea trenches, and arid deserts. Each of these environments is a treasure trove of undiscovered potential. Researchers are now looking beyond conventional sources, turning their attention to lesser-known bacteria and fungi that thrive in extreme conditions. These organisms have evolved their own defenses against microbial threats and may hold the key to unlocking new antibiotic classes. For instance, the search for antibiotics in the depths of the ocean has revealed remarkable discoveries. Marine bacteria and sponges have shown promise in producing compounds that can effectively combat resistant strains. One study highlighted the ability of a marine bacterium to produce a compound that inhibited the growth of antibiotic-resistant Staphylococcus aureus. It's a reminder of the untapped potential that lies in nature, waiting for us to unveil its secrets. In addition to seeking new compounds, researchers are also exploring innovative delivery systems. Imagine a world where antibiotics are delivered precisely to the site of infection, minimizing side effects and enhancing effectiveness. Nanotechnology offers such possibilities, allowing for the development of nanoparticles that can carry antibiotics directly to the bacteria causing the infection. This targeted approach not only improves treatment outcomes, but also reduces the likelihood of resistance developing, as the dosage can be optimized to be effective without overexposing bacteria to the drugs. Another alternative that has gained traction is phage therapy, a treatment that utilizes bacteriophages, viruses that specifically infect bacteria. This method stands in stark contrast to traditional antibiotics, which often affect a broad spectrum of bacteria, including beneficial ones. Picture a microscopic battle where phages seek out and destroy their bacterial targets, while leaving the rest of the microbiome intact. This precision is a game changer in the fight against resistant bacteria. Phage therapy has shown success in treating various infections, including those caused by multi-drug resistant strains. Imagine a patient suffering from a persistent infection, one that has evaded all conventional treatments. In such cases, phage therapy could provide a tailored solution. Doctors can isolate the specific bacteria causing the infection and identify the corresponding phage that will effectively target it. As these phages multiply within the infected area, they exponentially increase their numbers, leading to a focused eradication of the harmful bacteria. This therapy not only showcases an innovative approach, but also highlights the importance of personalized medicine in combating resistance. As we consider these innovative treatments, the significance of combination therapies emerges as a crucial strategy in our arsenal against resistance. The concept is simple yet powerful. By using multiple agents simultaneously, we can enhance efficacy while minimizing the likelihood of resistance developing. Imagine the potential of combining traditional antibiotics with newer agents, such as phages or even immune-modulating therapies. This synergistic approach can create a multifaceted attack on bacterial infections, tackling them from different angles. Combination therapies have already shown promise in clinical settings. For instance, studies have indicated that pairing antibiotics with phage therapy can enhance the overall effectiveness of treatment, leading to better patient outcomes. By disrupting bacterial defenses through multiple mechanisms, we can outsmart these resilient microbes. It's akin to a well-coordinated team of specialists working together to overcome a formidable challenge. Moreover, the importance of understanding the microbiome in this context cannot be overstated. Our bodies are home to trillions of microorganisms, many of which play vital roles in our health. When traditional antibiotics are administered, they can disrupt this delicate balance, leading to further complications. Combination therapies that take the microbiome into account aim to preserve beneficial bacteria while combating harmful ones. Imagine a therapy that not only targets infection, but also supports the restoration of a healthy microbiome. This holistic approach represents a significant shift in how we think about treatment. As we reflect on the journey of antibiotic development, it becomes evident that the future lies in innovation and collaboration. Scientists, clinicians, and public health experts are coming together to address the multifaceted challenges posed by antibiotic resistance. The urgency of the situation has fostered creativity and resourcefulness, leading to breakthroughs that were once thought to be mere dreams. The words of a renowned microbiologist resonate here. Science is a way of thinking much more than it is a body of knowledge. This philosophy drives the ongoing quest to uncover new solutions in the face of adversity. In the tapestry of modern medicine, the innovations in treatment stand as a testament to our resilience. As we navigate through the complexities of resistance, each new discovery adds a thread to the fabric of our understanding. The journey is ongoing, fueled by curiosity and a commitment to safeguarding human health. The potential that lies within the realm of antibiotic development is vast, and the innovations on the horizon could reshape the future of medicine. Our exploration of these advancements reveals not only the challenges we face, but also the ingenuity that propels us forward. The collective efforts of researchers and healthcare professionals serve as a beacon of hope, illuminating the path toward effective treatments that can combat even the most resistant strains. As we immerse ourselves in the stories of those pioneering these innovations, we are reminded of the profound impact that scientific inquiry can have on our world. The challenge of antibiotic resistance is formidable, yet it is not insurmountable. With each step we take into the future, we carry with us the lessons learned from the past. The journey of innovations in treatment is a testament to our ability to adapt and evolve in the face of adversity. As we forge ahead, we do so with the knowledge that the answers we seek are often found in the most unexpected places, waiting patiently for us to uncover their potential. Emerging from the shadows of traditional treatments, the quest for new antibiotics is not just a scientific endeavor. It is a race against time. As bacterial resistance continues to rise, researchers are tirelessly innovating to discover and formulate new antimicrobial agents. One exciting avenue of exploration is the potential of synthetic biology, where scientists design and engineer bacteria to produce novel antibiotics. Imagine a world where scientists can program microorganisms to produce compounds specifically tailored to combat resistant strains, effectively turning nature into a living factory of therapeutic agents. This approach, often referred to as bioengineering, harnesses the power of genetic manipulation to create bacteria that can produce antibiotics more efficiently. For instance, researchers can insert genes responsible for antibiotic production from one organism into another, enhancing yields and broadening the range of antibiotics available. This biotechnological advancement not only expedites the discovery process, but also expands the repertoire of potential treatments beyond what nature has traditionally provided. In the realm of alternatives to conventional antibiotics, the emergence of antimicrobial peptides is noteworthy. These small proteins, produced by various organisms as part of their innate immune response, exhibit potent antibacterial properties. The beauty of antimicrobial peptides lies in their ability to target bacterial membranes, disrupting their integrity and leading to cell death. Imagine a defense mechanism so effective that it can selectively target harmful bacteria while being harmless to human cells. Research in this field is still in its infancy, but the potential for these peptides to serve as a new class of antibiotics is immense. The exploration of plant-based compounds also offers promising alternatives. Many plants have evolved to produce secondary metabolites that exhibit antibacterial activity. From the ancient practice of herbal medicine to modern pharmacology, the use of plant extracts has long been recognized for their healing properties. By studying these natural compounds, scientists can identify and isolate specific ingredients that may serve as effective treatments against resistant bacteria. The vibrant world of botany, filled with colors, scents, and textures, holds secrets waiting to be unlocked in the battle against infection. In addition to these innovative approaches, the the importance of combination therapies cannot be overstated. This strategy leverages the strengths of multiple treatments to enhance efficacy and combat resistance. The rationale behind combination therapy is straightforward. When bacteria are faced with multiple agents attacking them in different ways, their chances of developing resistance diminish. Picture a multi-pronged assault where each treatment works synergistically, creating a robust defense against infection. One compelling example of this strategy in action is the combination of antibiotics with immune system boosters. By enhancing the body's own defenses while simultaneously targeting bacteria, this approach can lead to improved patient outcomes. Combination therapies can also involve the use of antibiotics in conjunction with non-antibiotic agents, such as adjuvants that enhance the activity of the antibiotic itself. This collaborative effort not only maximizes treatment effectiveness, but also serves as a buffer against the emergence of resistance. Furthermore, the rise of personalized medicine tailors treatments to the individual needs of patients, taking into account their unique genetic makeup and microbiome composition. Imagine a scenario where a patient's specific bacterial infection is matched with the most effective combination of treatments, minimizing side effects and maximizing recovery. This tailored approach is particularly relevant in the context of antibiotic resistance, where understanding the genetic profile of both the patient and the bacteria can inform treatment strategies. As we explore the landscape of innovations in treatment, it becomes evident that the fight against antibiotic resistance is not one-dimensional. It is an intricate interplay of science, creativity, and collaboration. Renowned scientist Paul Ehrlich once said, "The future belongs to those who prepare for it today." This foresight rings true as researchers, clinicians, and policymakers unite to prepare for the challenges posed by resistant bacteria. The commitment to innovation in antibiotic development is a reflection of our collective determination to safeguard public health. In this journey, the role of education and awareness also plays a vital part. As healthcare professionals and the public become more informed about the responsible use of antibiotics, the likelihood of resistance can be mitigated. Imagine communities empowered with knowledge, making informed decisions that promote antibiotic stewardship. This cultural shift can lead to a more sustainable approach to antibiotic use, preserving their effectiveness for future generations. The narrative of combating resistance continues to evolve, fueled by scientific inquiry and human ingenuity. Each new discovery and innovation serves as a reminder that while the challenges may be daunting, the potential for solutions is equally vast. The horizon of antibiotic treatment is expanding, and with it, the promise of improved health outcomes for individuals and communities alike. As we delve deeper into this intricate web of innovations, we are reminded of the resilience of both science and humanity. The collective efforts of researchers, healthcare providers, and the public illuminate the path forward, paving the way for a future where antibiotic resistance is met with a robust arsenal of treatments. The story of combating resistance is far from over. It is a dynamic narrative that continues to unfold, inviting us to witness the remarkable journey of discovery and hope. Chapter 8. Prevention Strategies: Reducing Resistance Development. In the ever-evolving landscape of public health, the role of antibiotic stewardship has emerged as a vital strategy in our collective effort to combat the growing threat of antibiotic resistance. This stewardship encompasses a range of initiatives aimed at ensuring that antibiotics are used judiciously and effectively, preserving their efficacy for generations to come. Picture a community where healthcare professionals, policymakers, and the public work together, each playing their part in a larger mission to safeguard our health. Public health initiatives are designed not only to promote responsible antibiotic use, but also to foster a culture of awareness and education. These initiatives often include guidelines for healthcare providers, encouraging them to prescribe antibiotics only when necessary and to select the appropriate type and dosage. Imagine a physician, equipped with the latest knowledge, considering the implications of their prescription. With every decision, they weigh the immediate needs of their patients against the broader consequences for public health. Antibiotic stewardship programs also emphasize the importance of monitoring and surveillance. By tracking antibiotic use and resistance patterns, health authorities can identify trends and develop targeted strategies to mitigate resistance. This proactive approach can be likened to a lighthouse guiding ships through treacherous waters, illuminating the path for healthcare providers and policymakers alike. As Dr. William Campbell, a prominent figure in antibiotic research, once noted, if we are to continue to treat infections effectively, we must understand the dynamics of antibiotic use and resistance. Hygiene and infection control measures serve as another cornerstone in the fight against antibiotic resistance. Imagine entering a hospital where every surface is meticulously cleaned, hand sanitizers are readily available, and healthcare workers rigorously follow protocols to minimize the risk of infection. These practices are essential as they help prevent the spread of resistant bacteria from patient to patient and within the broader community. The importance of hand hygiene cannot be overstated. Studies have shown that simply washing hands with soap and water can drastically reduce the transmission of pathogens. As we envision healthcare settings, let us appreciate the small yet significant actions that contribute to a larger defense against infections. These practices extend beyond hospitals. They are equally vital in schools, homes, and public spaces. The act of washing hands, a simple gesture, transforms into a powerful weapon against the proliferation of resistant bacteria. Infection control measures also encompass the proper use of personal protective equipment, or PPE, in healthcare settings. Imagine healthcare workers donning masks, gloves, and gowns, each layer serving as a barrier against infection. These measures are particularly crucial in environments where antibiotic-resistant organisms may thrive, ensuring that both patients and healthcare providers are safeguarded. Educational campaigns form another essential component of our strategy to combat antibiotic resistance. These campaigns aim to raise awareness among the general public about the responsible use of antibiotics and the risks associated with misuse. Consider the impact of a well-crafted message reaching thousands of individuals, encouraging them to complete their prescribed courses of antibiotics and avoid sharing medications. Public awareness campaigns harness the power of storytelling to engage communities. Picture a vibrant poster in a local clinic, depicting a family discussing the importance of only using antibiotics when necessary. These visuals can resonate deeply, fostering a culture of understanding and responsibility. When people comprehend the consequences of antibiotic misuse, they are more likely to take action, whether that means consulting a healthcare professional before seeking antibiotics or advocating for better hygiene practices in their communities. In addition to public awareness, educational initiatives targeting healthcare providers are equally crucial. These programs emphasize the importance of ongoing training and knowledge sharing among medical professionals. Imagine a gathering of doctors and nurses, exchanging insights about the latest research in antibiotic resistance, discussing case studies, and sharing successful strategies for effective patient care. Such collaboration enhances the collective expertise of healthcare teams, ultimately benefiting patients and communities alike. The integration of technology into educational campaigns has also transformed how we communicate about antibiotics. Imagine the power of social media platforms, reaching audiences at lightning speed with evidence-based information. These digital tools allow for real-time engagement and dissemination of knowledge, creating a dynamic dialogue about antibiotic stewardship. As we consider the various strategies at our disposal, it is essential to remember that reducing resistance development is not a solitary endeavor. It requires collaboration among healthcare providers, public health officials, and the community. Each individual plays a crucial role in this collective effort. In this context, let us reflect on the question, how can we as a society contribute to the preservation of antibiotics? The answer lies in our daily choices, from how we approach our health to how we support public health initiatives. By cultivating a culture of awareness and responsibility, we can collectively steer the course toward a future where antibiotics remain effective tools in our medical arsenal. The landscape of antibiotic resistance is complex, shaped by numerous factors that intertwine with our understanding of bacteria and their behaviors. As we have explored in previous chapters, Bacteria possess remarkable adaptability, allowing them to survive in various environments, even in the face of antibiotics designed to combat them. This reality underscores the urgent need for robust prevention strategies that address not only the immediate challenges but also the underlying factors contributing to resistance. In the grand tapestry of public health, the threads of education, hygiene, and stewardship are woven together, creating a resilient framework for combating antibiotic resistance. Imagine this framework as a shield, protecting our health and well-being while fostering a deeper understanding of the delicate balance between human activity and microbial life. As we move forward, let us hold on to the vision of a world where antibiotics remain effective allies in our fight against infections. By embracing prevention strategies and championing responsible practices, we can nurture a healthier future for ourselves and for generations to come. The role of public health initiatives in antibiotic stewardship extends beyond mere guidelines and recommendations. It encompasses a commitment to fostering a culture of accountability and shared responsibility among all stakeholders. Imagine a scenario where healthcare providers, researchers, and patients engage in a continuous dialogue about antibiotic use. This collaborative approach can lead to more informed decision-making, ensuring that antibiotics are prescribed only when truly necessary, and that patients understand the importance of adhering to treatment regimens. One could think of this as a community garden, where each participant contributes to the overall health of the ecosystem. Just as gardeners nurture their plants with care, healthcare professionals nurture their patients' well-being while simultaneously considering the larger impact on public health. This interconnectedness highlights the importance of public health campaigns that aim to educate not just healthcare providers, but also the general public about the responsible use of antibiotics. Hygiene and infection control measures form a critical barrier against the spread of antibiotic-resistant bacteria. Visualize the impact of a well-thought-out infection control protocol in a hospital, where patients are safeguarded through meticulous cleaning, isolation of infected individuals, and robust vaccination programs. These protocols are not just checklists. They represent a comprehensive strategy to create environments that significantly reduce the incidence of infections. In schools and workplaces, the promotion of hygiene practices can have a ripple effect. Let us consider how teaching children the importance of hand washing can shape behaviors that last a lifetime. By instilling these habits early, we are not just preventing immediate infections. We are also cultivating a generation that understands the value of hygiene in a broader context. Personal protective equipment, or PPE, is another vital aspect of infection control. Picture healthcare workers in full gear, serving on the front lines against infectious diseases. Each item they wear, masks, gloves, gowns, acts as a shield against pathogens, protecting both the wearer and their patients. During outbreaks, the significance of PPE is magnified, showcasing the necessity for proper training and protocols to ensure its effective use. Educational campaigns play a transformative role in shaping perceptions and behaviors surrounding antibiotic use. Imagine a vibrant community event where local leaders and healthcare professionals come together to discuss the importance of antibiotics in a friendly and engaging manner. This environment fosters open communication, allowing individuals to ask questions and gain a deeper understanding of the implications of antibiotic misuse. Effective campaigns often employ relatable narratives that resonate with people's experiences. When individuals hear stories of loved ones affected by infections that could not be treated due to antibiotic resistance, the message becomes personal. This emotional connection drives home the urgency of responsible antibiotic use, encouraging community members to advocate for their health and the health of those around them. The integration of technology into these campaigns has further amplified their reach. Consider the powerful impact of social media platforms in disseminating crucial information about antibiotics. A single post can inform thousands of people about the dangers of self-medication or the importance of completing prescribed courses. This rapid dissemination allows for real-time updates and guidance, creating a dynamic conversation about antibiotic stewardship that engages diverse populations. Moreover, educational initiatives must also focus on healthcare providers, ensuring they are equipped with the latest knowledge and best practices. Picture a continuous education program that brings together professionals from various fields, physicians, pharmacists, and public health officials, to share insights and collaborate on solutions. This collective intelligence fosters an environment where innovative ideas can flourish, ultimately benefiting patient care. As we reflect on the interconnected nature of these strategies, it becomes clear that the fight against antibiotic resistance is multifaceted. Each thread, public health initiatives, hygiene and infection control measures, and educational campaigns, intertwines to form a strong fabric of community resilience. This resilience is built upon the understanding that every individual has a role to play, whether as a healthcare provider, a patient, or a concerned citizen. How can we harness this collective strength to make a lasting impact? By fostering an environment where information flows freely and individuals feel empowered to make informed choices, we can create a culture of stewardship that extends beyond mere compliance with guidelines. The challenge of antibiotic resistance is not solely a scientific issue, it is a societal one. It calls for a concerted effort from all corners of the community, individuals, families, healthcare systems, and governments. Imagine a world where we approach our health with mindfulness and awareness, understanding that our choices have consequences for both ourselves and the greater community. As we continue to explore the depths of antibiotic resistance, let us remain focused on the proactive measures we can implement today. The cultivation of a culture of responsibility, supported by robust public health initiatives, hygiene practices, and educational outreach, will empower us to confront the challenges ahead. Together, we can work towards a future where antibiotics remain effective allies, preserving their power against infections that threaten our health. In this pursuit, we are not merely passive observers, we are active participants in a collective journey. Each step we take, each conversation we engage in, contributes to a larger narrative, a narrative of hope, resilience, and the promise of a healthier tomorrow. By embracing this vision, we can ensure that the legacy of antibiotics is one of strength and sustainability, rather than a cautionary tale of what could have been. Chapter 9. Future Challenges. Addressing Antibiotic Resistance. As we look to the horizon of the future, the specter of antibiotic resistance looms ever larger, casting a shadow over the achievements of modern medicine. Predictions suggest that the evolution of bacterial resistance will not only continue, but may accelerate in the coming years. Imagine a world where common infections, once easily treatable, become life-threatening due to the emergence of resistant strains. This scenario is not just a distant possibility, it is a growing reality that demands our attention. The mechanisms that underpin resistance are deeply rooted in the biology of bacteria themselves. As discussed in previous chapters, bacteria possess an extraordinary ability to adapt, a trait that is both fascinating and alarming. With each cycle of antibiotic use, those bacteria capable of withstanding treatment proliferate, while their susceptible counterparts diminish. This relentless cycle of survival and adaptation can lead to the emergence of strains that are resistant to multiple antibiotics, complicating treatment options, and increasing the burden on healthcare systems worldwide. Reflecting on the potential impacts of this phenomenon on global health systems, one must consider the implications for both individuals and society at large. Hospitals, often seen as sanctuaries for healing, may become battlegrounds against infections that resist every available treatment. The World Health Organization has warned that antibiotic resistance could lead to millions of deaths each year, a chilling forecast that underscores the urgency of addressing this challenge. Picture a healthcare system overwhelmed by patients suffering from infections that were once easily curable. The economic burden could be staggering, straining resources and diverting attention from other critical healthcare needs. Moreover, the ripple effects of antibiotic resistance extend beyond human health. Imagine the implications for global food security. In agricultural contexts, where antibiotics are routinely used to promote growth and prevent disease in livestock, resistant bacteria can infiltrate our food supply. The interconnectedness of our ecosystems means that the choices made in one sector can have profound consequences in another. The movement of resistant bacteria from farms to tables exemplifies the urgent need for a holistic approach to health, a realization that the health of humans, animals, and the environment are inextricably linked. Despite the mounting challenges, there remain significant research gaps that call for exploration. While we understand some mechanisms of resistance, much remains to be uncovered about the genetic and environmental factors that drive this process. What role do various ecosystems play in the development of resistance? How do environmental stressors influence bacterial behavior? By delving deeper into these questions, scientists may uncover new insights that lead to innovative strategies for combating resistance. Furthermore, the exploration of alternative treatment modalities is a crucial area needing further investigation. As we discussed, traditional antibiotics are becoming less effective, calling for a pivot toward novel approaches. Imagine harnessing the potential of phage therapy, where viruses specifically targeting bacteria are employed to eliminate infections. This innovative avenue could complement existing treatments, reshaping our understanding of how to combat resistant strains. In addition, the role of the microbiome in human health offers another fertile ground for research. Our bodies are home to trillions of bacteria, many of which play beneficial roles in maintaining health. What if we could leverage this knowledge to prevent infections or restore balance after antibiotic use? Such possibilities could lead to groundbreaking advancements in how we approach health and disease. As we envision the future, the collaboration between scientists, healthcare providers, policymakers, and the public becomes increasingly vital. The adage, "An ounce of prevention is worth a pound of cure," resonates profoundly in the context of antibiotic resistance. By fostering a culture of awareness and education, we can empower individuals to make informed decisions about antibiotic use, thereby reducing the selective pressures that drive resistance. Moreover, global cooperation is essential in addressing antibiotic resistance, as bacteria do not recognize borders. Imagine a world where countries join forces to track and combat resistant strains, sharing data and resources to enhance collective knowledge. This international effort could pave the way for effective strategies that transcend geographical and cultural barriers, ultimately benefiting public health on a global scale. As we reflect on the challenges ahead, it is clear that the path forward requires a multifaceted approach. The story of antibiotic resistance is still being written, and the decisions made today will shape the narrative for generations to come. The resilience of bacteria, while formidable, is matched by our capacity for innovation and adaptation. Together we can rise to the challenge, striving to safeguard our future against the threat of antibiotic resistance. What will this future look like? Will it be a world where we have successfully harnessed our understanding of bacteria and resistance to create a healthier society? Or will it be one where we face the consequences of our inactions? The answers to these questions lie within our grasp, awaiting our commitment to change, exploration, and collaboration. Envision a future where the race against bacterial resistance unfolds not just in laboratories, but in the streets, homes, and farms of every community. The prediction of evolving bacterial resistance is not merely a scientific abstraction. It is a reality that demands our immediate attention. As bacteria adapt to the antibiotics designed to eliminate them, we face an escalating cycle of resistance that could redefine the landscape of public health. What if we could forecast this evolution with greater accuracy? Understanding the genetic changes that confer resistance could empower scientists to anticipate and counteract these threats before they take root. The implications of antibiotic resistance extend far beyond individual health. Picture a health system straining under the weight of resistant infections. Hospitals, once symbols of healing, may become overwhelmed with patients suffering from infections that are resistant to first-line treatments. This burden on healthcare systems could lead to longer hospital stays, more complex medical interventions, and increased mortality rates. As the World Health Organization emphasizes, the potential for millions of deaths from antibiotic-resistant infections is a sobering reminder of the stakes involved. The economic ramifications are equally daunting. Imagine the ripple effects on healthcare costs, with increased expenditures on treatments, prolonged hospitalizations, and the need for advanced therapies. Countries with limited healthcare resources may find themselves particularly vulnerable, struggling to provide adequate care while facing the specter of resistance. The delicate balance between treating infections and managing healthcare costs could be disrupted, resulting in a healthcare crisis that transcends borders. Moreover, the interconnectedness of our global food systems introduces further complexity to the antibiotic resistance narrative. In the realm of agriculture, the widespread use of antibiotics in livestock raises pressing concerns. Imagine a farm where antibiotics are routinely administered not just for illness, but also to enhance growth and prevent disease. These practices can lead to the emergence of resistant bacteria that find their way into the food supply, creating pathways for transmission to humans. This stark reality underscores the necessity of a comprehensive approach to health, one that recognizes the intricate links between human, animal, and environmental health. As we contemplate the future, we must also confront the significant research gaps that exist in our understanding of antibiotic resistance. While we have made strides in identifying some mechanisms of resistance, many questions remain unanswered. What genetic and environmental factors are at play in the development of resistant strains? How do different ecosystems contribute to the proliferation of these bacteria? Addressing these gaps is essential if we are to devise effective strategies to combat resistance. The exploration of alternative treatment modalities offers a promising avenue for research. As traditional antibiotics wane in effectiveness, scientists are looking beyond conventional methods. Picture a future where phage therapy gains prominence, a treatment that harnesses viruses that specifically target bacteria to combat infections. This approach not only offers a potential solution to resistant infections, but also encourages a more nuanced understanding of our microbial world. Could these viruses work alongside traditional antibiotics to create a multifaceted defense against bacterial threats? Additionally, the role of the microbiome is a burgeoning area of research that could transform our approach to health. Our bodies harbor a complex community of bacteria that play essential roles in maintaining health. Imagine harnessing the power of this internal ecosystem to prevent infections or restore balance after antibiotic use. By understanding how the microbiome interacts with pathogens, we could develop innovative strategies to reinforce our defenses against resistant bacteria. Collaboration emerges as a cornerstone in addressing antibiotic resistance. The challenge is too great for any single entity to tackle alone. The adage that prevention is better than cure rings particularly true in this context. By fostering a culture of awareness and education, we can empower individuals to make informed choices about antibiotic use. This shift toward responsible medication practices can help mitigate the selective pressures that drive bacterial resistance. Global cooperation is equally vital. Imagine a world where nations unite to share data, resources, and best practices in tracking and combating antibiotic-resistant strains. Such collaboration can transcend borders, enhancing our collective understanding and response to resistance. Countries can learn from each other's successes and challenges, pooling their expertise to forge effective strategies that safeguard public health on a global scale. As we gaze into the future, the narrative of antibiotic resistance is still being shaped. The decisions made today, whether in laboratories, hospitals, or farms, will reverberate for generations to come. The resilience of bacteria, though formidable, is met by our collective capacity for creativity and adaptation. The journey ahead is fraught with challenges, yet it is also rich with potential for innovation and collaboration. What role will each of us play in this unfolding story? Will we be passive observers or active participants in the fight against antibiotic resistance? The answers lie in our hands. awaiting our commitment to explore, innovate, and collaborate in pursuit of a healthier world. As we move forward in addressing the multifaceted challenge of antibiotic resistance, it becomes increasingly clear that our approach must be both innovative and inclusive. The future of our health systems depends on a deeper understanding of the biology behind resistance mechanisms. Imagine scientists delving into the genetic blueprints of bacteria, unlocking the secrets that allow these microorganisms to thrive despite the presence of antibiotics. This exploration can unveil new targets for drug development, offering hope for treatments that can outpace bacterial evolution. One promising field lies in the study of bacterial communication, known as quorum sensing. Picture bacteria as members of a community, capable of sensing their population density, thereby coordinating their behavior to enhance survival. This fascinating mechanism not only allows bacteria to thrive, but also can contribute to their resistance. By understanding how these communication pathways work, researchers could disrupt the signals that enable bacteria to mount collective defenses, potentially rendering them more vulnerable to treatment. Furthermore, the importance of diagnostics cannot be overstated. Rapid and accurate diagnostic tools can revolutionize how we prescribe antibiotics. Consider the scenario in a busy clinic, where a doctor is faced with a patient exhibiting symptoms of an infection. With the right diagnostic tools, the physician could quickly ascertain whether the infection is bacterial or viral, tailoring treatment accordingly. This precision can minimize unnecessary antibiotic prescriptions, reducing the selective pressure that fosters resistance. Exploring the realm of vaccines presents another exciting frontier. Vaccination offers a proactive approach to preventing infections, thereby decreasing reliance on antibiotics. Imagine a world where vaccines are developed not only against common bacterial pathogens, but also against strains known for their resistance. Such advancements could significantly lower infection rates, preserving the effectiveness of existing antibiotics and safeguarding public health. The agricultural sector must also undergo a transformation. The current practices surrounding antibiotic use in livestock can no longer be sustained. Envision farms where animal health is prioritized through alternative measures, such as improved husbandry practices, vaccinations, and probiotics. By reducing the routine use of antibiotics, we lessen the chances of resistant bacteria entering the food supply, ultimately benefiting both animal and human health. Education plays a pivotal role in this endeavor. As we cultivate a culture of awareness around antibiotic use, we empower individuals to make informed choices. Picture community workshops where families learn about the responsible use of medicines, the importance of adhering to prescribed regimens, and the necessity of completing antibiotic courses. This knowledge can foster a sense of shared responsibility, reinforcing the idea that every choice matters in the fight against resistance. Global health organizations are increasingly recognizing the need for a unified approach to combat antibiotic resistance. Imagine nations collaborating on research initiatives, sharing data and resources to tackle this issue collectively. Such partnerships could facilitate the development of global surveillance systems, enabling the monitoring of resistance patterns and the effectiveness of interventions. The strength of collaboration lies in its ability to transcend geographical boundaries, creating a network of knowledge and support that enhances our collective resilience. As we stand at this crossroads, it is evident that the challenge of antibiotic resistance is not insurmountable. The journey ahead demands creativity, commitment, and collaboration from all sectors of society. Each action taken today, whether in the realm of research, policy, or education, contributes to a more robust defense against the looming threat of resistant bacteria. The potential for a healthier future rests not only on the advancements made in laboratories and clinics, but also on the collective will to embrace change and foster a culture of stewardship. What kind of legacy do we wish to leave for future generations? Will we look back and recognize the moments when we chose to act decisively? Or will we regret opportunities lost? The answers to these questions lie within our grasp, beckoning us to rise to the occasion and forge a path toward a more sustainable and healthy world. Together, we can harness our knowledge and creativity to ensure that the story of antibiotic resistance ends not in despair, but in hope and resilience. Chapter 10. A Call to Action. Collective Responsibility in Antibiotic Use. As the sun sets over the horizon, painting the sky with hues of orange and purple, a sense of urgency fills the air, a call to action that transcends borders, cultures, and individual lives. The reality of antibiotic resistance is not just a challenge for scientists and healthcare providers, but a collective responsibility that beckons involvement from every corner of society. Imagine a world where common infections become untreatable, where the simple act of a minor surgery carries the weight of risk, and where our medical achievements are threatened by invisible foes. This is not a distant future, but a pressing concern that requires our attention now. Global cooperation is paramount in the fight against antibiotic resistance. Picture nations coming together, sharing insights and strategies, much like a network of roots intertwining beneath the surface of a vast forest. Each country's unique experiences and solutions hold the potential to enrich the collective understanding of this crisis. The World Health Organization has called for a unified response, emphasizing that no single country can tackle this issue alone. As we consider the interconnectedness of our world, how can we foster a sense of shared responsibility among nations? One path forward lies in collaborative research and data sharing. By pooling resources, countries can enhance their surveillance systems, identifying resistance patterns and tracking outbreaks with greater precision. Imagine a global database, a living document that captures the evolving nature of bacterial resistance, allowing for timely interventions and informed policy decisions. This collective effort can lead to a more resilient healthcare system, one that anticipates challenges rather than merely reacting to them. Engaging healthcare providers is another vital component of this strategy. Picture a physician standing at the crossroads of patient care and public health, where every prescription carries the weight of consequence. Responsible antibiotic use is essential in curbing the tide of resistance. Physicians must adopt the mindset of stewardship, recognizing that their decisions extend beyond individual patients to influence the health of communities. The greatest medicine of all is to teach people how not to need it, said Hippocrates, highlighting the profound responsibility that comes with healing. Training and education programs can empower healthcare professionals with the knowledge and tools they need to prescribe antibiotics judiciously. Imagine a vibrant hospital where medical staff engage in continuous learning, discussing antibiotic protocols and the importance of diagnostic testing before treatment. With every conversation, they reinforce the concept that antibiotics are not a one-size-fits-all solution, but a powerful resource that must be wielded with care. Moreover, fostering a culture of accountability among healthcare providers can lead to a paradigm shift. Consider a scenario where peer reviews and feedback mechanisms become commonplace, encouraging doctors to reflect on their prescribing habits and to learn from one another. This collaborative environment can nurture a sense of shared purpose, as each healthcare provider recognizes their role in the larger narrative of antibiotic stewardship. However, healthcare providers cannot carry this burden alone. Engaging the public is essential in creating a comprehensive approach to antibiotic resistance. Imagine individuals empowered with knowledge, understanding the implications of antibiotic misuse not just for themselves, but for their families and communities. Public awareness campaigns can serve as a potent tool in fostering responsible antibiotic use. These campaigns can educate the public about the importance of completing prescribed courses, the dangers of self-medication, and the need to consult healthcare professionals before demanding antibiotics. As we consider the role of the public, it's essential to ask ourselves, how can we inspire proactive participation in antibiotic stewardship? Stories of success can be instrumental in this endeavor. Picture a community that rallies together, sharing anecdotes of how responsible antibiotic use has led to healthier families and neighborhoods. By amplifying these narratives, we can foster a sense of pride and responsibility, encouraging individuals to become advocates for change. Schools and educational institutions also play a pivotal role in shaping attitudes towards antibiotics. Imagine children learning about bacteria and antibiotics, not as abstract concepts, but as real-world issues that impact their lives. Integrating antibiotic education into curricula can empower the next generation to be informed consumers, instilling in them the understanding that their choices matter. Furthermore, the food industry must participate in this collective responsibility, with agriculture being a significant contributor to antibiotic use. Imagine farmers transitioning towards more sustainable practices that prioritize animal welfare and public health. By embracing alternative methods for disease prevention in livestock, such as improved hygiene and better management practices, we can reduce the reliance on antibiotics. This shift not only benefits animal health but also safeguards human health by minimizing the risk of resistant bacteria entering the food chain. As we move forward, it is crucial to recognize that the fight against antibiotic resistance is a marathon, not a sprint. Each step we take towards fostering global cooperation, encouraging responsible use among healthcare providers, and engaging the public brings us closer to a future where antibiotics remain effective. Imagine standing at the edge of a vast ocean, each wave representing a small action taken collectively, shaping the shoreline of our health landscape. In this journey, we can draw inspiration from figures in the field who have championed antibiotic stewardship. Their commitment to preserving the efficacy of these medications serves as a guiding light. The words of one celebrated microbiologist resonate deeply. The only way to solve the problem of antibiotic resistance is to act together. This sentiment encapsulates the essence of our mission, a call to action that urges us to unite in purpose and spirit. As we reflect on our role in this narrative, let us cultivate a mindset of collective responsibility. Each of us, regardless of our profession or background, has a part to play in ensuring that antibiotics remain a cornerstone of modern medicine. Whether it is advocating for responsible use, participating in public education, or supporting policies that prioritize sustainable practices, every action counts. In this intricate web of life, the interconnectedness of humans, animals, and the environment underscores the importance of our efforts. Imagine a world where the threat of antibiotic resistance is diminished, where infections that once plagued humanity are kept at bay through responsible stewardship. This vision is within reach, but it requires the commitment of us all. The time for action is now. Let us embrace this collective responsibility, transforming awareness into proactive engagement and ensuring a healthier future for generations to come. Together, we can turn the tide against antibiotic resistance, creating a world where the gift of antibiotics continues to save lives and enhance health. The path forward in the journey against antibiotic resistance is illuminated by the necessity of fostering collaboration on a global scale. Imagine a vast conference hall filled with representatives from diverse nations, each bringing unique perspectives shaped by their experiences with antibiotic use and resistance. They gather not merely to discuss statistics, but to share stories of personal encounters with the devastating effects of resistant infections, creating a tapestry of human experiences that transcends borders. Through these conversations, we can begin to understand the nuances of antibiotic resistance, recognizing that it does not discriminate. It touches lives in every corner of the globe, from the bustling cities of developed nations to the rural villages of developing countries. This shared narrative must inspire collaborative efforts in research, policymaking, and public health initiatives. By learning from one another, we can develop targeted strategies that address the specific challenges faced by different populations. In healthcare settings, the role of education and training cannot be overstated. Imagine a world where every healthcare provider is not only aware of the importance of antibiotic stewardship, but is also equipped with the skills to implement it effectively. Continuous professional development programs can serve as vital resources, ensuring that clinicians stay abreast of the latest guidelines and evidence-based practices in antibiotic prescribing. Moreover, mentorship programs can play an essential role in shaping the next generation of healthcare professionals. Picture experienced practitioners guiding young doctors through the complexities of antibiotic treatment, sharing insights on when to prescribe, and when to withhold these powerful medications. This exchange of knowledge can foster a culture of critical thinking and responsibility, as practitioners learn to balance patient care with the broader implications of their prescribing habits. As we engage healthcare providers, we must also be mindful of the systems within which they operate. Hospital administrations can implement policies that promote accountability and transparency in antibiotic prescribing. Imagine a hospital where data on antibiotic use and resistance is openly shared among staff, creating an environment where everyone feels responsible for the choices made in patient care. This collective awareness can lead to more conscientious prescribing practices and ultimately contribute to a reduction in resistance rates. Yet, to truly tackle antibiotic resistance, we must extend our efforts beyond the walls of healthcare facilities and into the communities we serve. The public's role is critical, and a proactive approach to antibiotic stewardship must engage individuals in meaningful ways. Public health campaigns should aim to demystify antibiotics, providing clear messages about their purpose and the importance of using them responsibly. Consider a vibrant community event where health educators interact with families, sharing stories and facts about antibiotics. Imagine children participating in interactive sessions that highlight how antibiotics work and why completing a course of treatment is vital. These engagements can empower individuals to make informed decisions, fostering a culture where the community collectively values responsible antibiotic use. Furthermore, the role of social media and digital platforms cannot be overlooked. In an age where information spreads rapidly, harnessing these tools can amplify awareness about antibiotic stewardship. Picture a viral campaign that encourages individuals to share their stories of responsible antibiotic use, creating a ripple effect that inspires others to follow suit. By utilizing platforms that resonate with diverse audiences, we can cultivate a sense of community responsibility around antibiotic use. In addition to public engagement, the food industry must play its part in combating antibiotic resistance. The practices of antibiotic use in agriculture have far-reaching consequences for both animal and human health. Imagine farmers adopting rigorous standards that prioritize animal welfare without the over-reliance on antibiotics. By implementing alternative methods such as improved nutrition and biosecurity measures, the need for antibiotics can be significantly reduced. This shift is not only beneficial for the animals, but also serves to protect human health. Picture a world where the food supply is free from the threat of antibiotic-resistant bacteria. As consumers become more aware of these issues, they can drive demand for responsibly sourced products, encouraging farmers to adopt sustainable practices. The power of consumer choice, when aligned with public health goals, can act as a powerful catalyst for change. As we reflect on the challenges ahead, it is essential to recognize that the responsibility for combating antibiotic resistance lies with each of us. Imagine a global movement where individuals, healthcare providers, and policymakers unite in a common cause, fostering an environment that prioritizes health and well-being. The complexities of this issue may seem daunting, but together, we can navigate through the intricacies of antibiotic stewardship. The words of a renowned public health advocate resonate deeply. The greatest public health challenge of our time is to ensure that antibiotics remain effective. This statement encapsulates the essence of our collective mission. It urges us to take action not just for ourselves, but for the future of medicine and public health. As we continue this journey, let us remain vigilant and committed. The call to action is not a distant echo. It is a resounding invitation to engage, educate, and empower one another. Each step we take in promoting responsible antibiotic use contributes to a healthier world, where the benefits of these life-saving medications can be preserved for generations to come. Together, we can forge a path towards a future where antibiotic resistance is no longer a looming threat, but rather a challenge we have collectively overcome.